आगे लगाइए, ऐसे ऐसे एक मिनट लगे, उसको पीछे करो।
A very good morning to all the esteemed guests, faculty members, research scholars, and students. I welcome you all to the second day of the International Conference on Advances in Cancer Biology, Prevention and Therapy. It is an honor to have the speakers we have on board today, who through their talks and discussion will highlight the key points, trends, and reforms in the evolving field of cancer biology. It will indeed be an honor to hear and learn from their rich experience. Further, we have also organized a paper and poster presentation opportunity for our delegates to put forward their reforming thoughts in this vast field. Now, for the current session, I would like to introduce our chairpersons, Professor Gyazuddin Khan, and co-chair, Professor Dinesh Raj Modi. Professor Gyazuddin Khan is a forensic medicine expert and currently Joint Director at UP State Medical Legal Cell, Lucknow, India. He has completed his MBBS degree and later qualified as MD in the field of medicine. Now he is pursuing PhD in forensic medicine. He has delivered lectures in numerous conferences and seminars at universities, national and international level. Also, he has published numerous research papers in several national and international journals. He has been selected 12 times as a medical officer in the Hajj mission. He has received a Lifetime Achievement Award from Indian Congress of Forensic Medicine and Toxicology. I would now like to request our chairman, sir, to kindly present the memento to Professor Khan. Thank you, sir. This session will be co-chaired by Professor Dinesh Raj Modi. He is currently designated as a professor in the Department of Biotechnology at BBAU, Lucknow. He completed his postgraduate from GB Pant University of Agriculture and Technology, Pantnagar District, Nainital. He had completed his PhD from University of Hyderabad in 1991. He has a training experience of 36 years and a research experience of 41 years, and has research articles published in over 35. He has his expertise in biotechnology and applied microbiology. I would now like to request the convener of the conference, Dr. Tabish sir, to kindly present the memento to Dr. Modi. Thank you, sir.
Now, I would like to introduce our first speaker for the session, Dr. Shrikant Kumar Rath. Dr. Shrikant Kumar Rath is a chief scientist and professor at Division of Toxicology and Experimental Medicine, CSIR and CDRI. He completed his PhD in certain anti-fertility agents on the reproductive organs of the male laboratory mouse from Banaras Hindu University, Varanasi, and postdoc from Center for Cellular and Molecular Biology, Hyderabad, India. He was a Raman Research Fellow during 2007 to 2008 at Bloomberg School of Public Health, Johns Hopkins University, Baltimore, Maryland, USA. He was deputed as toxicologist to Center for Safety Assessment for Food and Feed, New York, USA. He was a scientist in charge, Academic Unit, CDRI, January 2013 to 2015. He is a member of Distinguished Committees of Government of India. Now, I would like to invite Dr. Rath for his talk.
बोलना यूज చేయాలి right?
No, sir. So the previous slide, next slide, and this.
ये लाइट्स ऑफ कर देंगे ये दोनों Good morning, everybody. I am grateful to Tavis and his team in Ramswarup University to invite me to deliver this talk. I am extremely happy to see Dr. D.R. Modi and Dr. Khan to chair this session. Let me at the outset tell Dr. D.R. Modi is one of the oldest collaborator, and we had a common mad-like student whose PhD we did with very much difficulty with Gorakhpur University. Today what I am going to speak about is actually the phytochemicals, and I'll put this question to you all. Are they really control cancers? You must be knowing that there are several cancers in our body. As the concept goes in cancer, each and every cell has the potency to develop into cancer, to develop into tumor. But we have hundreds of cancers which are very frequently happening in our body, and it happens in animals also. But since animals are not ready to tell you, they don't speak about their pain, so we are concentrating in our own cancer. Okay, so there is a great challenge. It is one of the killer disease in the globally, and there are a lot of labs working on this. More than a century, actually 1920, the first report in cancer treatment and its horrific development in the human race is being described. But even after a century, we have no answers to cure cancers. Although in some cases like breast cancers, we have some attainment. We have achieved something to control the cancer if it is discovered at the early stage. But if it is late stage in third or fourth, then it is really difficult to control the cancer. There is no family perhaps in the world which is not have a bad taste of cancer, because some or the other person of the family members, it's not going there, dies out of cancer, and all of our organs, mostly vital organs like breast, liver, lungs, prostate, all are getting affected with cancer. So there is a chance we should try for new avenues to see that whether the cancer can be controlled. We have several successful drugs I will show in the slides, but still people are believing that phytochemicals can be a wonderful drug for the treatment of the cancer. Please, next slide. Those who are students, that building what I was showing in this slide is actually of new CDRI. Those who have not visited CDRI, they must visit there. And I am very happy to see there are several students here working in this organization. They are from IRTR or CDRI, and all are smiling faces in the first or second rows. And this is a gratifying moment for a teacher or a scientist to see their own student are flowering in different organizations. And in the morning while I was talking with the Vice Chancellor, I told him that when you have people from CDR and IITR, you don't have to bother for anything. You just give them support, they will grow your institute. I think I am very confident about that. And this picture shows that how serious is the disease. Those who are sitting in the back may not be able to see clearly, but it says that actually how much happening in the males and females, and what are the types of cancers in the body, and how many people are actually affected expected in 2020, and how many they are being killed. Around 19 million people they actually get affected every time, and males and females are in different death categories, but nevertheless both in females or males the cancer is very, very killer. And around 50% of the cancers, next slide, around 50% of the cancer affected people are dying actually. In the breast, it may be around 35-40%, whereas in lung cancer, more than 80% people they die. So it is certainly a matter of gravity how it is actually people are affected and how they are dying. Nowadays in India, the cervix cancer and the cancer of lungs and liver is very in around Lucknow it is very getting peak actually, and I don't know what is the reason. Maybe some of the environment and the water conditions may be the cause for these cancers. Next. So there are several wonderful drugs are there. Most of them are working certainly, but every one drug doesn't fit to everybody. So doctors take the combinations, and I shall be speaking about what are our results from our lab. And there are also phytochemicals, alkylating agents, antibiotics, antimetabolites, and other compounds which are actually used. For example, etoposide, it's a plant phytochemical. So all these drugs are available to the doctors, but still there is a need because the death rate is increasing and we are unable to control the disease. Next. So you can see from vincristine, you can see all these drugs, etoposides, all these are being tested by paclitaxel, one of the most popular drug. All are from the plant sources, and similarly we are planning to get researching actually whether we can increase this kitty so that people can be benefited. Next. Next. So basically every plant, wherever you go, you will see the plants, and every part of the plants, they have the phytochemicals. Since we have also our biochemicals inside our body, similar the plants have their own, but plants have many because plant has to sustain itself, right? It has to take its own, make its own food. It has to survive in a different environment, and it has to protect itself. And these are most of the secondary metabolites from the plants, and in different chemical groups they can be categorized. And these are not the only list of the groups, but several other groups are also there. Only people who are concentrating in getting the chemicals out of this are actually belonging to these groups. Next. So phytochemicals are the biological functions people have tested in worldwide in most of the labs, and our interest of today is anti-cancer. Most of the carotenoids, polyphenols, curcumin and flavonoids, and they are all inhibitors of tumor, inhibitors of development of lung cancer. There are all evidences existing in the literature to find out. They are also can be in antibacterial, they can be detoxifying agents and other. So people think of that all these chemicals, in addition to other chemicals, whether they can be advantageous for some of the cancers. Next. So actually every day we are taking some chemicals through our food. Okay, there maybe every sabji, wherever it is from Bhandara or in our home or kitchen, we take curcumin, right? Lot of curcumin. Yesterday you might have taken in Bhandara, but still people think that we are taking regular Bhandara, we are Hanuman Bhakt, but still cancer is affecting us because of the reason is curcumin we are taking in a raw stage, and curcumin has a very bad habit of not getting into your body. It goes to your alimentary canal and goes out. It doesn't go to your blood systemic circulation so easily and so frequently and so much. So that is bioavailability in pharmaceutical terms we tell. The bioavailability is very less, 3 to 4%, which is actually not a desired material. That's why curcumin, in spite of its very anti-inflammatory effect, anti-ischemic effect, anti-cancer effect, we are unable to make it a drug which can be available in the market. Okay, in the country like companies like Cadila, they have developed some curcumin formulations for daily ingestion for keeping you fit, but still that cannot be utilized as a anti-cancer drug because of the reason of the bioavailability. Every morning, if you are taking with milk the curcumin, it actually, if you take some grams, hardly any microgram quantity, it goes to the systemic circulation. Therefore, it is unable to get the benefit. We are unable to get the benefits out of it. Next. So prevention is better than cure. Therefore, I would advise all the students, young people, it is better instead of burger, pizza, and all these marketed materials which are available outside the college campus or university campus, you better go for veggies. You can take all these. If you go to United States of America, you can see the people are taking these veggies very frequently. All leaves, all types of leaf, kacha they used to take raw, right? That just wash and take, right? And we are not habituated that because we are habituated to masala and talna wala cheese that we like very much. That's why perhaps we are unable to take. But if you are taking regular vegetables and fruits and raw materials, maybe your cancer progression will be less, occurrence will be less, and of course progression will be delayed, and you can get the better benefits out of that. Next. So people have tried in all these 8 type of cancers using different cell lines, and these are the cell lines of different nature, and people have tried to find out all these chemicals which are very, very popularly utilized in different labs. Mostly they are epigenin, codenine, epicatechin, all these are camphorol, resveratrol, all these are people are using, and they have demonstrated that these cells are being dying after the treatment. But when it translates into the in vivo conditions, that is in in animals, it is not happening. So how it is happening here? Okay, so that is a challenge actually, how you will translate from these small molecules from the in vitro results to the in vitro. In vitro it works in 10 micromolar or 15 picomoles. Somewhere it works in different labs, but it doesn't work in the animals where we are actually have to give the more quantity, whether injectable or through the mouth. Next. So the challenge is to difficult find a new drug as per our requirement. Our requirement today is we have several, but we do not have options, and it has to have a better efficacy. But if there is no better efficacy, then there is no chance of getting a new drug into the all regulators in any country will ask actually whether the drug is effective better what is existing today. One. Second, they will ask whether the toxicity is less or not, because this is great challenge. Most of the around 25% of the patients actually they die out of the toxicity than the efficacy actually. Many people they don't respond, and they are affected so much by toxicity of the compound, so that they cannot respond to efficacy, and toxicity risk that balance is lost and people die. So, and the third thing doctors are they want in the clinic is fit to all. So one drug for all types of cancer. It doesn't happen. If you go to the hospitals, you will see that different doctors are trying to do lot of permutations and combinations to give the best benefit to the patient. So these are the 3 challenges we have in front of us to treat the patients. So basically we try to think of that none of these epigenin or catechin or camphorol or curcumin, they will not be single-handedly will be a drug for anti-cancer, because developing this as a drug is a very, very challenging as of bioavailability and pharmacokinetics problem. Then specificity is a problem. So therefore we thought that drug combination in chemotherapy we can actually check to find out whether it can be against the drug resistance, or it can be cancer stem cells against, or metastasis, toxic effects chemotherapy. So toxic effects, of course, very well designed toxic studies are being there everywhere, and we can very easily do the things in the lab. But the greatest challenge is cancer stem cell and metastasis, because you do not know where the cell has gone and sitting in other tissue, and wherever it wants, it will grow up and make cancer. So it comes like a morning dew you do not know. Morning you get up, you see the all plants in different leaves, the dew spots are there. Similarly, exactly in the body, the cancer tumors will be developed, and together they will grow to big tumors, and patient dies because it requires lot of energy. Since it requires lot of energy, it takes lot of blood, and the blood, whole blood, whatever you have, the tumor uses that, and whatever oxygen in your blood, the tumor uses, and make all the venation, and the blood vessels are being within the tumor to supply the oxygen, and rest of the organ or the rest of the body is actually devoid of oxygen, and the patient dies. So we have to take these challenges, and without this, we thought that the advantage of combination will be widens the therapeutic index and maximizes the efficacy. That means therapeutic index means if it is 15 milligram per kg body weight, the dose is, can we reduce it? The moment we reduce it, the efficacy without compromising the efficacy, actually we are reducing the toxicity. That is the challenge that widens the therapeutic index. Then maximizes efficacy. Naturally, the efficacy should not be compromised in any case. Then can hit more than one critical molecular target, because you know that the cancer is a multi-gene disease. Actually, it's a syndrome, and a lot of P53, in addition to P53, lot of genes are being affected, and that gene actually makes you very disturbing, because lot of genes, lot of targets, and one molecule cannot go there and bind the receptors and bring the effects. So that is the reason why we thought of, then purpose of to characterize the chemopreventive agents that have a minimal toxicology and most efficacious in nature. Next one. So we did experiment around 10 years back, that is actually to increase the cyclophosphamide efficacy with resveratrol. You can see here in the cyclophosphamide, and here is the cyclophosphamide, only resveratrol. It has some inhibitory effects, but not much like the cyclophosphamide. But when you combine, there is some effects, and toxicity goes down, and you can see actually the cyclophosphamide dose reduce like anything when you are adding the resveratrol. So we did a long experiments and could find out the resveratrol can be available easily and can be treated to the patient with cyclophosphamide with a reduced dose, so that to bring down the toxicity. We did the same thing here also in other thing. So this picture actually is the normal cells. So the effects are, you can find out the normal cells, some cells will be dying, but other things are very comparable in both these cells. There is a human cell, is a dog cell, and there are hardly any differences in three groups. But in this next slide, you can find out, you see the moment you are changing the, these are 4 quarters you can see, white, all these are cyclophosphamide combinations from 1 micromole to cyclophosphamide 50 micromole, and in 200 microliter of resveratrol, you can get actually 97.28% of the success. So majority of cells will die. But in cancer, you remember, the cancer cell will just behave like a parasite. If that 2.7% around, if they are living, so they will be the cause of the metastasis, right? Like a resistant parasite. Okay? You must be knowing recent COVID incidences, how the Omicron, then 156, 157 beta type, all these are coming and moving around the environment, right? They are getting mutated and getting resistant, okay? And your all vaccines will fail. And we do not know whether India will get back in another wave or not. Because of this 3%, they will be notorious enough, because they all know all these doses and drugs. So they will adjust themselves very smartly, that beyond our thinking, and they can make havoc. So one has to be very careful. So I will not recommend that this is the dose which can be, but it is achievable. We can do that for if you are increasing the doses. Next. So again we try to do the similar experiments with MDMB, which is a breast cancer cell, and you can see in this resveratrol alone and resveratrol plus CPM, there is some advantages where these 15 micromole doses can do wonderful in comparison to the 25 micromole of only CPA. So doses can be reduced and they can bring effects in comparable to the controls. Next. We can we showed the apoptotic MCF7 cells. You can see the cells, and here also apoptosis rate points. This shows in this particular combination 10 micromolar CPA and resveratrol 100 microliter. One can find out in G phases 10 micrometer MCF and 100 micrometer resveratrol, we can induce enough apoptosis to kill the cells. But still, if there are green cells somewhere, that means it is not 100% done, so we have to still further titre and find it out. But beyond that we tried, but we could not able to achieve such things what we could able to get in this particular doses. Then we did the cyclophosphamide resveratrol in the combination doses, not bring better effects to HepG2 cells in a different cell lines, that is actually a liver cell, and almost similar effects, a little better effects of course, which is in comparison to cyclophosphamide, we got a better effects here, and some of them are actually not at all inhibited in certain doses. That means there is still a scope to increase the titration and make the concentration perfect. So, but 15 micromole of the CPA and 100 micrometer of resveratrol giving some better rates, but we are target, let me tell you, it should be the target, not beyond that. Okay, so there is no compromise in case of cancer cells for compromising with the data. Then we checked actually G1, G stage and see the apoptotic peak whether it can be, and apoptotic peak we are finding out again in the CPA resveratrol better than the previous only resveratrol or CPA. Well, just remember the cyclophosphamide has to be comparable without compromising the anything. But you see, you can find out the significant effects when you have CPA and resveratrol together. So we studied the caspase activity. You must be knowing, those who are studying biology and biochemistry, they must be knowing the caspase activity as more essential for controlling the tumor. And we actually induced the caspase activities with all these inducers as by substrates. They are all substrates for caspase 3, caspase 8 and caspase 9. Then we find out that caspase is being activated in these cells, and we measure that, and it shows that the cells can go to apoptotic stage in both the direction, whether both the way of apoptosis. Next. So the conclusion is resveratrol increases the efficacy of cyclophosphamide, but at the same time it is not actually the ultimate what we have. We are focused to find it out. We could not able to do that. Similarly, we started doing the bioactivity compound of the therapeutic efficacy of celicoxib and colon cancer. This is another cancer. I showed the data of the breast and liver. Now I will show you the effects of natural bioactive compounds, two bioactive compounds in colon cancer. I will not go into details of the colon cancer, but I would wish to tell that there is some defects in the genetic materials starting from P53, APC, KRAS, WNT signaling and other things, and from the normal colon it becomes the abnormal colon, and slowly the cells will grow without control and becomes the tumor and become the headache for you, and that has to be controlled. And similarly, as I have already described, the colon cancer has got a very powerful segment of the population where it is. And around Lucknow and PGI and KGMU and the Kant Hospital, the colon cancer patients are increasing. Around 29% of the cancer patients in Lucknow are actually belonging to the colon cancer segment, and they come from Raebareli and Pratapgarh, this area. Maybe somewhere this metallic toxicity in the environment or water is maybe the causative, I don't know, but it may be the causative agent for this cancer. And we tried with celecoxib. You must be knowing that celecoxib is selected COX-2 inhibitor, and NSAID has been approved by the US FDA for adjuvant treatment of the familial adenomatous polyposis, and that is the reason why we consider this drug, because this is already approved, and with this approval can we go forward with a phytochemical, and that the limitation of course is there in the cardiovascular risk associated with the long term usage and poor bioavailability. As I have already told for curcumin, this also has a very poor bioavailability. We thought that whether it can be increasing the bioavailability or not, and at the same time whether it can be treated as a anti-cancer drug. So the objective is evaluation effects of selected natural bioactive compounds and mechanistic studies and in vivo validation whether it works in the animals. So this you people must be knowing, in everybody's kitchen it will be there. What is this? क्या बोलते हैं उसको? बच्चे, ये चित्र में क्या दिखाई दे रहा है? यस यस, काला मिर्च, राइट, काली मिर्च. ये फीमेल जेंडर है शायद. काली मिर्च है, और ये काली मिर्च का पाउडर और सेलिकप्सिक का पाइपेरिन के साथ वी स्टार्टेड डूइंग रिसर्चिंग, and we found some interesting results, and basically we started studying the in vitro studies, and with that we did all the mechanism studies, and ultimately we checked in Balbisi mice in a xenograft model and find out some results, and that I will be sharing. That co-administration of piperin, this kali mirch actually piper langa, right? Piper langa se, sorry, langa kya hai? What is that? I forgot its name. Specific name. That PIP is actually piperin, and enhances the relative bioavailability of celecoxib. So it increases the bioavailability. That once I told the concentration bioavailability in your body, if it is increasing, then efficiency is expected to come to target the disease. So you can see there is a increase. There is an increase in the bioavailability celecoxib alone, which is blue, and it increased when you treat with piperin. Not much, but still the increase is there, and it is comparable. The more amount, the increase for 25 hours, that means the more amount of available piperin and celecoxib is in the body. So that will help you to actually target the disease, that is tumor. Okay. So that is the reason why we did this experiment, and we found out that the PIPT leads to automation of rhodamine, the accumulation of rhodamine ST29 cells. That means it actually, both the things in the cells that will be available for a long time, and it accumulated the moment. Sorry for these pictures, we are unable to see the things, but here there is accumulation PIP, and in comparison to that little more is there, you can see in these slides. Then we did this selectivity inhibitor of the growth of human colon cancers, and you can see in the celecoxib and piperin there is significant decrease, and that means it is somewhere it is increasing the killing factors, that is IC50 of 50 is there, and piperin is here, but together they can kill more cells in these particular cells, that is IEC6 and SC29. Then we did actually celecoxib in combination of piperin synergistically inhibited growth of ST29 cells. You can see how it has gone celecoxib here, but the growth down here with PIP, and that shows that synergistic effect is there, where we can have this results. So many more, around one is additive and synergistic effect are observed in this. You can find out in this table how dose reduces reduction index, you can find it out, and in combination index you can find it out, and you can see the 0.8842, and this is 0.86, little increase is there. We did also Western blotting to see the expression of synthesis down regulator COX-2, because you have COX-2 inhibitor, celecoxib. So we found it is acting fine. Then with piperine and celecoxib, you can see here the reduction of the protein in comparison to these. Celecoxib and piperine actually combination alters the morphology also. That means this is making the cell abnormal, and while the cell is dying, it actually becomes abnormal, and if it is contaminated, it becomes abnormal. So there is a visual proof that it alters the morphology, and in the same cell actually combination induces externalization of the this is all about the mechanism, how it must be working. And pre-treatment with NAC reduces co-treatment induced. We treat also, pre-treat the cells with NAC, then again treat with these compounds, and we found out the suggesting ROS mediated induction of apoptosis by combination. So it actually works through ROS mechanism, ROS pathway, and these are some of the others observations. What I wanted to show is celecoxib and piperine activates the caspase cascade of ST29 cells. So caspase cycle, it is getting affected. So you can see actually how it is here in celecoxib and piperine together, and they work in coherence and improves here and protect the cells. That means killing the cells. That means it maybe the combination will work perfectly all right. So similar, it also we found out the oxidative state in this particular cell ST29 cells. Then the combination and mitochondrial membrane, whether potentiality is affected or not, and we found out that it is working in the, you can see in the BAX and BCL2, this is our apoptotic pathway proteins, and they are getting improved because of this together treatment, whereas only celecoxib actually you can compare this improves actually. So that means it is killing somewhere mitochondria membrane potential, and the celecoxib stream the properties of colon cancer cells. Again, these similar cells in the we checked, and somewhere we found out that here celecoxib and piperine together are actually reducing the OCT4 and affecting the cells and killing the cells significantly in comparison to celecoxib alone. We checked also whether the pathway WNT beta catenin signaling pathway is getting affected or not. We found that it is getting little encouraged the pathway, and it is killing, maybe that is the reason more than one pathway it is acting actually, and you can see the significant difference celecoxib and piperine together, and see significantly it goes down the cell numbers. We then went to in vivo animal models. Just let me know what is the time, okay? Just 5 minutes before you I will stop, right? So similarly we did in the animals, and you can see the tumor getting reduced when you are putting the celecoxib and the PIP. We took the cancer cells and injected into the animals, and then the tumor grows. Then we get the treatments. You can find out the growth of a control tumor and how it is being reduced. Celecoxib alone and piperine alone. But here from this picture, if you compare actually, you can see the piperine is working better than celecoxib, but together they are working wonderfully. But ultimately it has to reduce. But the major problem is that the animals after bearing such tumors survive very less. So we have to experiment within the days, so that we can see the tumor is going. When the tumor goes completely, the animal doesn't actually live up to that death. And if you are leaving that, maybe the animal will be killed, and you will conclude that maybe due to tumors effects, the animal is dying. So that is why we are killing the animals much earlier than its death, and find out whether these tumors are being, this the histology, you can find it out, how the histology is in these tumors. So the conclusion is combination works fine synergistically, and it works actually through different methods of controlling the cell death. And similarly, we also did another experiments with this material, people must be knowing this is dalchini, right? So that is cinnamaldehyde. Again, we tried with celecoxib, and almost similar results we find out. We did this cytotoxicity. You can find it out. This cells STC11C 3 cells, and you can find it out how the 55, and these are the concentration celecoxib alone and cinnamaldehyde, and together they get beautifully drop the things into this. It's really actually in different cell lines very significantly this combination works. Similarly, the previous one, and you can see the cells celecoxib and cinnamon co-treatment induces the apoptosis in these cells. You can see the cells in concentration of that, and lot of cells are being dying in these cells, and very significantly actually it reduces the cell numbers. Similarly, in different cells, STD116, similar results we have found out, and we did this. Thank you. We did this similar experiments with FACS and find out the mitochondrial membrane potential, and we found the significant results. You just see the corner blocks, it is significantly different from the celecoxib alone, and only cinnamon. So this shows that combination therapy can work fine, and invasion of colon cells also we tried to check to find out that whether this compound can work in the metastasis or not. So you can see the results here significantly actually together it reduces much significant actually. It reduces the metastasis. So maybe drinks of kadha of this cinnamon may work in cancer patients to help your normal treatment and give advantage to the patients. So I will not go into details of the mechanism as the time doesn't permit me, but we extremely worked in this two molecules, and that first part is actually this also we tried to do under hypoxia conditions and see that because hypoxia is one of the essential elements in the cancer tumor development, and you can see the significant decrease in the cells. So again the similar experiments we did, and we can find out, you can see actually the celecoxib and the cinnamon combination actually reduce the tumor from this level to this level. So with this, the conclusion is this also works synergistically, and such compounds can be further pursued to see whether it can be druggable or not. So once we are talking of druggability, so we have to do a lot of experiments, and we have to do actually most of the synthetic things around the molecules, so that semi-synthetic molecules you can develop to increase the bioavailability and decrease the toxicity and get the desired results. So recent work in 2022, two people, Hemlatha is Indian and Mendia, both together actually published a paper in Applied Biochemistry. They have actually goes systematically, very systematically, and find out 50 phytomolecules. All are very common type, and they first started with the Lipinski's rule. Lipinski's rule of 5 is nothing but there are rule of 4. Lipinski gave a formula that all the orally taken drug actually should have a minimum small size, that is 500 Dalton, less than 500 Dalton molecular weight, and then not more than 5 hydrogen bond donors, or not more than 10 hydrogen bond acceptors. Then log P should not be more than 5. This is rule of 5, and all these 50 molecules they tested also in binding energy comparable to standard drugs, and they did pharmacokinetic activities. So actually they did the reverse way, whether it can be a druggable molecule or not. So it's a good approach, I think, in my opinion, and I am very impressed to see that they compare with the standard drugs which are actually available as anti-cancer drugs, and all these 11 molecules, you can see this binding energy is almost comparable, and further studies are being done. They also depicted the amino acid involved in the hydrogen bonding. So all these things are well calculated. Further, in vivo, this is actually an in silico study. In vivo studies they have to do, and they find out whether any of them, all these 11, any of them can be a drug or not. So with this, I would conclude my lecture today, and I am grateful to my colleagues, Dr. Vivek Garg, who works in a LCI, Lucknow Cancer Institute, my great friend. Then Muhammad Wajuddin, who did all the pharmacokinetic studies. Now we have lost him. He has joined a laboratory in England. Then Dr. Saras Sharma, my great friend, who is our chair. And then Neetu Singh is actually one of the postdoc, very initial cyclophosphamide experiments she did. She was in KGMU and not, of course, she is now no more there. Then Sonal Srivastav is a scientist in ICMR Institute. He is my PhD student, and now flying to US Tampa for joining a postdoc. Jayant is working in a private company as a scientist in charge in cell biology. He just completed 2018. Sakshi Misra is a postdoc still working in the cancer, skin cancer. Prabhas is another postdoc who is working as a scientist in KGMU. Divya Tandon again working as a scientist in KGMU. Aman has joined recently to a university who is going to submit his thesis, and he's actually worked very hard to find out the angiogenesis in cancer. Then Gaurav is working, and Sonam is working in prostate. Thank you so much for inviting me again. Thank you organizers for giving me
Thank you, sir, for such an interesting talk. The session is now open for question and answer.
Hello. Thank you very much, Dr. Rath, and we are really delighted with your presentation. One thing I will just query, how you have combined the garam masala, that is the black pepper and javitri, all these things. How the idea came to your mind in treating cancers, colon cancer, GI tract cancer?
You rightly said, sir, garam masala, right? Wherever there is masala, we used to be attracted, right? Whether it is a movie or it is a masala in the kitchen, right? So masala, actually it came from literature. People are doing some, as I have told, that phytochemicals are everywhere, and people are working on this lycopenes and other things. Lot of literature you will find, but we found that these things are rather little unexplored, and these are, people take them as kadha, right? So that is as they are taking as drinks. Do you know? Then during this COVID period,
Yeah.
a director of ILBS came to our institute and he declared that please don't make it official, but people are taking kadha so much that they have lost their liver. Okay?
Yeah.
So that video you can see in the YouTube. So anything is too much is too much. So we tried our best in a conservative way. So whether there is an advantage or not, okay, our aim is if it can be worked in the in vivo experiments for humans directly, so the life expectancy will be increased, right? In the cancer, generally doctors think that 5 years life expectancy is increased. That means there is a success. You must be knowing that, right?
Oh, that
So-
is the, excuse me, that is the very good prognosis you told along with. Why I just asked you, because black pepper, it causes hemorrhoids.
Right.
It is one of the causative agent in hemorrhoids. Some people who are taking black pepper, kali mirch in their food, so that is the very much toxic for the lower GI tract, that is the hemorrhoids. But as per your study, it is very good, and it is giving synergistic effect in treating some colonic growth. So thank you very much. Thank you, sir.
Sir, again for your question I will answer. So, as a toxicologist, we always think dose and duration, okay? Anything, you take everything which is overdose, there is bad effect. Whether it is alcohol, it is water, or it is even water, people have died. There is a report in, from Australia. Apple, 160 apples, Casseret book, it is written, people have died. So it can happen. And as you have told, everything, they have black pepper we take in a, we are only talking of piperine, okay? There are several other phytochemicals there, and many, they may be accelerating the toxicity. And whether it is, for example, metformin, one of the most safest drug, people take 1 gram morning and evening, right? Still, if you take more, you will be affected. So there is nothing which is not toxic. Everything is toxic. As a toxicologist, if you tell me everything is toxic, you have to take the dose and duration. Both the things has to be very, very carefully controlled. So that is why we have put a series of molecules in milli, micromole and picomole conditions.
No,
so that we can control.
that's right. Actually,
And our purpose is, if in the in vitro and in vivo we will get the controlled dose, then automatically will translate into some tablet of 20 mg, 30 mg. That will be, I think it will be okay.
Sir,
but if it is
actually,
beyond that, it may be.
Actually, I am also medical toxicologist.
Right, sir.
My degree is Doctor of
Forensic
Medicine, Forensic Medicine and Toxicology, as you know.
Right, sir.
So I am very much interested in toxicology. For the kind information to these students, nicotine we are taking,
Yes.
that is in low dose, it is the brain stimulant.
Absolutely.
But if 60 milligram of nicotine is taken by sublingual, it is the cardiac arrest.
Absolutely.
It is the fastest killing poison in the world. That is because we are getting myth, cyanide is the poison which kills, but not. The fatal period for cyanide is 30 minutes. And for nicotine, if 60 milligram in liquid form we take, that is the fastest killer. In fraction of second, our heart stops. Thank you very much, sir, for your kind and very, very informative, and you can say it is good for research for the coming generation. And over to my friend, Mr. Modi, please.
Yes, Dr. Raj, thank you very much for your informative, and what I not say informative, it was more of an educative one. Okay. Now to the students, as Dr. Rath has said, the dosage and the duration of drug. Okay, that is very important. Now, coming at this point, just I want to take, whenever a doctor prescribes a medicine, okay, you will take the medicine and all that. Just notice some of the medicine strips have got a red mark. What does it indicate? Any idea? Not all the tablets. Some of the medications, okay, the strip of that medicine will have a horizontal stripe or a red.
Schedule H drugs.
Why it is there? Not for all. For some. Okay. See, Dr. Rath has indicated that has gone through that, and that then struck my mind, okay, to tell you. That is there, if you want to go to the detail, it says, this should be taken on advice of a medical practitioner, okay, and the duration will be there. If it is continuous, that is dose and duration, okay, that means you continue for a long time, then it will be a toxic.
Right, sir.
See, this is another thing, but we normally, we don't realize why it is there. Arey bhai, kuch hoga, lal hoga, something to trademark or to sell or something, nothing. Okay, there is some scientific concept over that. So what I want to tell to the students, preferably wherever I go, okay, you should have self-questioning in yourself. Okay, whatever it is, wherever it is, okay. And every product, when it comes from the company and all that, they'll give the instruction, everything will be there. But we hardly look into and read that. Now, being a student of the science and the modern technology and all that, that should be your approach. Okay. Why not this? Why not? Okay. Okay, now not to take much of time, a simple question I want to ask. Now there is one term called as a pulse polio. Why it is pulse? If you are immunized, then you take the second dose, then you say it is a booster. Then what is this pulse? कभी आया आपको क्वेश्चन है ना? Why it is pulse polio? Now, second, for just to have thinking, all the vaccines which are being administered are injected, but not the polio. It is oral polio vaccine, orally to be taken. किसी को कभी क्वेश्चन गया दिमाग में कि क्यों हम ओरल ले रहे हैं और दूसरे ऑल आर इंजेक्टेड? Okay, put this question to your mind, you will get the solution. Okay, okay, and I will give the answer immediately, but I don't want to have like something like a Maggie, है ना? ये करना, and then everything go to हाँ सब समझ में आ गया ना? I want students to think it over there. Okay, why it is there? Self questioning होना चाहिए, है ना? Then only the things will. That is the thing. Okay, that's the reason why I say education. Okay, literate. Okay, literacy day we celebrate. Okay, to know the things. Okay, very good. Then that, what is? That is literacy means learning with understanding. Is a real literacy. Not to write your name, to sign something like that will not be a literacy. Okay, that you should know, realize why it is, and question yourself. So with this, I will take leave. Sorry for taking this time. Okay, just because Dr. Ratha rightly pointed out the dose and the duration. Okay. लास्ट में मैं कहना चाहता हूँ हिंदुस्तान में हर आदमी एक डॉक्टर है, सबसे बड़ा देश है. कहीं कुछ अरे कुछ नहीं, ये ले लो. अरे there is a reason why, and in my university we have got a health center and all that. There is doctor to take any medication. Just call him, and he is so courteous. At any hour of the day, I call him. डॉक्टर साहब ऐसा सा प्रॉब्लम क्या लेना है, बस ये डोज लेना, एंड देन आई राइट द नेम ऑफ द मेडिसिन एंड गो टू द शॉप. द अदर डॉक्टर इज द शॉप ओनर. अरे साहब ये नहीं है, इसका सब्स्टिट्यूट ये ले लीजिए. आई सेड नथिंग. देन अगेन विल कॉल डॉक्टर, सर ही इज गिविंग दिस सब्स्टिट्यूट. देन ही लुक्स एट, नहीं ये मत लीजिए, हमने जो लिखा है वही लीजिए. ठीक है. इन सब केसेस इसे अच्छा आप सब्स्टिट्यूट ले सकते हैं. सो दैट because that medical shop owner, a person sitting, he is interested in sales. He is not interested or worried about your health. Okay, he wants money. So unknowingly you are damaging your own self. Okay, that's the reason why one should be very. That was a very alarming line told by Dr. Rath, that is the doses and the duration. It is very important because we'll be getting, because what he has designed. Then Dr. Khan has asked me, what made you to go for this Kali Mirch and Dalchini? Okay, ये सब चीजें हैं. So this is how the things will come. That is what we say, what science is. Okay, with this I'll take your leave. Sorry for that.
Rath sir, I have, so it was a very informative and knowledgeable lecture. I've been listening you since last so many years, since I was your PhD, one of the PhD student in that institute. But I have one more thing to say, that as I'm heading agriculture here, and we have one term which is known as traditional indigenous technical knowledge, that is ITK, and I've gone through a number of papers which were published by Ayush Mantralaya, but they don't have that particular logical and scientific backup like you said in your lecture that round about this much amount and this much dose will be injurious and this much will be beneficial. So I think it will be very beneficial for the human healthcare if we combine that particular findings which we have in our historical background with the logical backup and scientific background. So I really congratulate for taking this up, the role of phytochemicals in human healthcare.
Thank you so much.
Thank you so much,
Actually,
sir.
let me tell this August audience that Ayush Ministry actually have become empowered after Modi government, okay? So lot of activities are being going on, and the only one agenda Mr. Modi told to the ministry is you put your drug in international market, okay? So that is the only one line he told. That one liner actually made everything very different there. Ayush now is, Ayush has many institutes, 37 institutes, CCRS. But all of them are all actually practicing that Veda format without knowing basic things. So they have forced us to, Modi ji told after visiting CDRI that you go and talk CDRI and then use them. That's the language actually. So after that, lot of communication are being made on behalf of CSIR, Dr. Reddy, who is the CDRI director and IIIM director, and me and Dr. Vishwajanani. Traditional knowledge, we have a TKDL actually, traditional knowledge development, like digital library actually. All three are actually member from CSR side, and from that side again another Srikanth is there who is DDG, and Ayush ministry head, some Acharya has joined now, and there is a Ayush AIMS, All India Institute of Medical Science, Ayush in Delhi. So there Nesari, Madam Nesari and her husband, all these we have a joint group, and every week actually we had meeting. Yesterday also I had meeting. So now scientifically how we can prove that our traditional knowledge is working, we have to prove that. So all this toxicity battery has to be done, everything we have planned, chalked out, and if it works fine, then we can actually land something in the international market. You see how marketing Ramdev ji does. That marketing we CSR people are not doing. We are unable to do because we are basically scientists. So now it is lot of culmination is happening. I think maybe your dream will come true. I will advise if you have that IKDL park and something. This for all phytopharmaceuticals and Ayurveda things. Captive cultivation is must, because we get lot of arsenic, lead and other things which are unwanted things in the plants. Okay, so that has to be taken care of, and that can be taken care of by the people like you who are knowledgeable and knowing the environment and this quality of plant and quality of sand and mud and other things. I think things will come up, and since I came to know now, then if you have some 20 acres land and other things, we can also try to cultivate some. Yes, CMAP actually does with many farmers in Raebareli and throughout India they do. In this Lucknow area, Raebareli actually we have taken to culture Ashwagandha, 118.
Sir, I'm happy to share that recently my department has signed an MOU with Dr. Rajput. He is running his own NGO. He is also from CMAP only. And what he does is he works on medicinal plants only, and we are planning to join our students in that venture and inculcate this particular feeling in the farmers of the local area to shift from the normal crop rotation of wheat and paddy towards medicinal farming. And contractual farming is coming up very nicely in the adjoining areas. So we are already working on that, sir.
Very good.
I would now like to request Professor Khan to kindly present Dr. Rath with the memento. Thank you, sir. There's a small announcement to be made. All the participants of oral presentations are requested to submit their presentations. Thank you.
Moving on with the session, I would now like to invite Dr. G. Sunil Babu, who is Associate Professor in the Department of Biotechnology at Baba Saheb Bhimrao Ambedkar University. And his area of expertise is cancer genetics. So over to you, sir.
Good morning to everyone. Respected Chairpersons Modi sir and Khan sir, and distinguished scientist Rath sir, and the faculty members of SRMU and the students. First of all, I thank the organizers for giving me the opportunity to share my laboratory work with you all the people, especially Dr. Tabish. So actually this conference, my today's topic is role of human cytomegalovirus UL148D on ER stress-induced apoptosis. So first question is how this topic is suitable to the cancer-related conference. Yes, the thing is, in the cancer is a global burden, and 12% of the cancers were caused due to the viruses. So recent studies and literature indicates that human cytomegalovirus is also going to cause the cancers. So earlier, this was not considered as an oncovirus like Epstein-Barr virus and also human papilloma virus. But this virus in recent literature is showing the oncogenic nature. So what our lab is doing is, so we are working since 10 years about the functional validation of these viral encoded microRNAs. The thing is, previous lecture we came to know about the apoptosis, how this apoptosis was utilized by the cancer or tumor cells. So in this talk, we are going to study how this HCMV virus is going to exploit the apoptosis pathway for its survival and thereby causing the carcinogenic potential. Okay. So human cytomegalovirus is such a virus, it is omnipresent. So you will wonder to know that 100%, 99% of the human adult population in India is positive to this virus. So if I am going to show, so how the, okay. Okay. Oh, if you are going to show, okay, the ratio, India is going to have the 95% of the population, adult population shows the seroprevalence. So the thing is, if we are having the above 95% of the population, why we are not going to get any clinical symptoms is, this is a beta herpes virus, and all the herpes viruses are going to have a characteristic feature of giving the long latent periods. So how this virus is going to give the long latent period if the apoptosis is working fine? So these viruses is a special character of this virus. It is having a lot of its genome dedicated to support the immune response as well as the apoptosis. So our work is, we want to study the microRNA's role because the viral microRNAs actually have the potential because most of the people had worked with the proteins, and the UL36 to 38 genome of the HCMV is dedicated for the subversion of the apoptosis. Now what we are thinking is even microRNAs are also going to have a role in subverting the apoptosis. So as you already know, because this is a virus and virus microRNA functional validation is difficult. So we divided our studies into the in silico studies as well as the in vitro studies. So we have done some bioinformatic tools and identified what the probable HCMV microRNAs and what the probable cellular apoptotic gene targets. And after that, we have taken the model cell in vitro studies where we are going to validate our functional predictions. So what we have done is we have taken the apoptotic pathway genes, and with the help of the RNA hybrid and RNA22, these are the bioinformatic tools which are going to be used to find the mRNA and microRNA bindings. And among them, we had seen many pathways, but today topic, I am going to restrict myself to the ER stress induced pathway, because we had done other pathways also, that is mitochondrial dependent pathway also. But today we are going to limit to the ER stress induced apoptosis. So our results shows one interesting molecule, that is endoplasmic reticulum to nucleus signaling molecule. So this molecule is going to be targeted by the HCMV microRNA. Now, so what we studied is, so we have a microRNA, that is the HCMV mir UL148D, and the target gene is going to be the ERN1. So then these in silico predictions we are turned into the in vitro studies, where we have taken the HEK293 T cells as a model, and we induced the apoptosis through the staurosporine, because it is a classical chemical which is used for inducing the apoptosis in the cells. And this apoptosis inhibitions and evaluations are going to be studied through the microscopy, flow cytometry, and caspase 37 assay. As Radha sir had already discussed about these things. So what we have done is, okay, so the slide is not that much clear, but anyhow, what I am going to show you all is, here we have seen the cells, and these are the HEK293 cells, and once we are going to treat the cells with the staurosporine, you are going to see the apoptotic nuclei. This is the DAPI staining, and this is the propydium iodide, and this is the combined. So when we treat these cells with the HCMV microRNA, the apoptotic nuclei, the nuclear blebbing is going to be reduced. Then what we thought is, okay, this we have seen the reduction. Then what we thought is whether this reduction in the nuclear blebbing and apoptotic nuclei is due to our microRNA or not. So we used the inhibitor of the microRNA, and we found that the nuclear blebbing is reversed. That means whatever the nuclear blebbing is initiated through the apoptosis was reduced is due to the HCMV microRNA. So then what we have seen is, then we have taken to the flow cytometry data. And here also what we are going to observe is, again, the HCMV microRNA mir UL148D is going to reduce the apoptosis caused by the staurosporine. So this we have seen. Then again, what we have seen is, we again evaluated this one with the caspase 37 assay. So Promega is going to give the another assay where we can see the effector caspases, caspase 3 and 7, whether this caspase 3, 7 levels are going to be decreased or not. Because these many studies we have done, because we have taken the microRNA, and microRNA is whether it is going to inhibit or not, to make ourselves sure that this microRNA is actually inhibiting the apoptosis. We have done these many assays. Now, so the point is, as you already know, how this microRNA is going to downregulate the apoptosis, because you all know the microRNA function is, it is going to bind to the 3' UTR regions of the mRNA and downregulate the mRNA degradation. So what the mRNA is going to be targeted? So this is the question. So as I already told you, because in our in silico predictions, predict that ERN1 is going to be the functional target for this microRNA. So we have seen the ERN1 mRNA quantitation through the qRT-PCR, and found that when we are going to see that this ERN1 molecule is going to be reduced when we are going to use the mRNA mimic. So that means we made sure that in silico predictions, and also we confirmed that apoptosis inhibition, and here these results also shows that yes, when we are going to give the mir UL148D, the mRNA of ERN1 is going to be decreased. So then what we have done is, yes, we have taken the whole UTR region, And with the help of the cloning studies, we cloned this UTR region in the PZX expression vector. And this we are procured from the gene copia, where we have the whole site, UTR region, as well as the deleted site of this UTR also we have seen. And the results shows that, and this is called dual luciferase reporter assay. So this dual luciferase reporter assay will says that whether the mRNA is going to bind to the mRNA or not. So for that reason, we have with the lumino-luciferase activity we have seen, and these activities shows that when we are going to treat with this mimic, the luciferase activity is down-regulated. When we are going to block the microRNA function with the inhibitor, the luciferase activity is increased. When the UTR region is deleted, binding site is deleted, and there was no effect in the luciferase activity. This confirms that our microRNA is able to bind to the 3' UTR region of the ERN1. Now, so we had gone. So further, whether this microRNA binding, yes, we are confirmed that. Yes, we confirmed the microRNA is binding to the UTR region of the ERN1. Whether this binding translates to the repression of the protein or not. So we checked with the Western blotting, and with this Western blotting, we are going to see, yes, there is a if it is there, the ERN1 protein is going to get down-regulated. This shows that ERN1 is going to be down-regulated. You may be confused that why we are putting here is IRE1 alpha. Because ERN1 is the gene, and the protein encoded is going to be called as the IRE1 alpha. So then we have seen with the same thing with the, okay, SIR microRNA we are doing, because another work also we had simultaneously initiated because we are all doing. So what is the effect of the siRNA for this one? So we designed the siRNA for the 148D, and we have seen in 148D also we are going to see the degradation of the, down regulation of this protein. So this clearly shows that yes, the microRNA is going to inhibit the ER1 stress inducing molecule. So then, very quite interesting, how this IRE1, it is an ER stress sensing molecule, how it can down-regulate or how it can affect the apoptosis. This is the question. So IRE1 performs two functions. One is, it is going to do the, it is very interesting, this RNA splicing. IRE1 protein is going to do the splicing of the XBP1 mRNA. So actually we wondered, XBP1, how it is going to get untranslated, spliced. The spliced product of XBP1 is going to be a transcription factor, and it will goes to the nucleus and produce the ER-associated degradative enzymes. So any stress is there, so XBP1, IRE1 is going to get activated. If IRE1 is going to get activated, XBP1 is sliced, and XBP1 spliced product is a transcription factor and go and bind and activate the genes which are responsible for the enzymes degradation of the ER unfolded or proteins. So we studied the XBP1 levels, and we observed that the spliced one and unspliced one also we studied. So that means when we treat with this microRNA, so IRE1 level is also decreased, and IRE1 levels are decreased. The splicing of the XBP1 and XBP1 splicing is also get affected. So then another pathway is also there. If IRE1 is there, IRE1 is also do another one. Suppose ER stress is there. So in first phase, XBP1 is going to be, splicing is going to occur. If this XBP1 splicing ka wajah se, if it is not able to resolve the ER stress, then it will also induce the apoptosis. This is the point. So in this way, this IRE1 molecule with the JNK phosphorylations, it will turn the pathway to the apoptosis. So this is called ER-induced apoptosis. So we have seen the JNK phosphorylations also, and observed that these JNK phosphorylations are also reduced. So what we are hypothesized is, yes, during the ER stress, so what is going to happen is IR1 is going to get ER inactive, but due to the stress, IRE1 is going to get activated. So once IRE1 is going to get activated, it is going to splice the XBP1. This is the one model. So second one model hai, if the ER stress is unresolved, so what will happen? The IRE1 is going to active with the help of the TRAF1, and it causes the PJNK means phosphorylations and induce the apoptosis. So HCMV virus, how it is stopping the apoptosis is, it is going to stop the apoptosis, ER stress apoptosis, basically by translationally repressing the IRE1 protein. This is our study's result. So if we see the pathophysiology, so yes, when the HCMV is going to interact in the cells, then the ER stress is going to come, and this ER stress is going to be unresolved by the first pathway, that is the XBP1 pathway. Then this was shifted to the another pathway, that where the IRE1 with the help of the TRAF2 increases the phosphorylations of the ZNK and causing the and inhibiting the ER-induced apoptosis. So ER stress-induced apoptosis inhibition, how the virus is going to get benefited is, because you already know in regulation of the means apoptosis always beneficial to the viruses. In the first time they are going to means modulate in a such a way, the outcome of the response is going to be helpful to the virus survival. So the studies, our summary of the study is HCMV 148 regulates the ER-induced apoptosis, and the functional targets for this microRNA is ERN1 or IRA1 alpha. So these are the publications where we have done, we have shown from these studies, and I thank the means my funding agencies DBT and DST, and also our university BBAU, and also the research collaborators Sanjay Yadav, CSIR IITR, now he is in AIIMS Raebareli, and my research team Abhishek Pandey, Rajkumar Kalpo, and Aarti Priyadarshini. So thank you for your patient listening.
Thank you, sir, for such an engaging talk. The session is now open for questions from the audience. Okay, moving on, I would now like to request Professor Modi to kindly present Dr. Babu with the memento.
Excellent lecture.
Thank you, sir.
Moving on, I would like to introduce our next speaker for the session, Dr. Feroz Khan, who is a senior principal scientist and professor at CSIR CIMAP. He has completed his doctoral in UP Technical University in bioinformatics. He completed his postdoc in pharmacology and toxicology from SCAC School of Pharmacy and Pharmaceutical Sciences, University of California, San Diego, USA, and PhD in bioinformatics from UP Technical University, Lucknow. His research specialization is in biotechnology, which includes computational molecular biology, regulatory genomics, and in bioinformatics, which includes cheminformatics and drug discovery. Now, I would like to request sir to please proceed with the talk.
So previous slide, next slide, please.
Okay. So good morning, all of you. I'm thankful to the organizing committee members that they have given the opportunity to deliver a talk on this international conference. As you know, my topic is related to virtual screening of important anti-cancer agents, especially the natural compounds. So what the approach basically we used to screening out the potential candidates. So the most important approach internationally used in lead identification and optimization. As you know, the drug discovery stages are of different level, and the first lead-based approach is basically the lead identification optimization method. And after that, we go forward for target identification and target validation. And after that, we go for preclinical and clinical studies. So the very first step is to find out the potential agents. So how you can find out these agents? So the first approach mostly used by worldwide researchers is QSR approach, which is a traditional approach and classical method and mainly used by the medicinal chemist. So we can define it, QSR is a quantitative structure-activity relationship. So you see the definition, it's a relationship of biological activity of the compounds which is previously studied or characterized through experimental approach with their chemical properties. So these chemical properties basically called as descriptors or the identifiers. So there is structure is, if you see the structure of anti-cancer agents, they are of different level. Structurally, they are diverse in natures, but still they have a common feature which is anti-cancer activity. So why these compounds have anti-cancer activity? We don't know actually. So there is some relationship within the structural properties which is hidden inside, and our approach, this QSR approach, is basically to extract out this feature through relationship studied. So there are lot of computational methods, but we use the machine learning methods to screen out large number of compounds and find out the patterns among the phytomolecules, natural drug compounds, and other analogs compounds with their properties. This relationship studies covers two important parts. One is identification of the compounds for training the model, and then identification of the chemical features, which is also called as feature selection method, which required important selection of correlated features which have relationship with the biological activity. So if you see the FDA, US FDA website, they have strictly mentioned that before any approval of the compounds, for active compounds as a drug molecule, you should at least submit their structure-activity relationship studies. So I highlighted some of the component. So uncircled part is especially highlighting the structure-activity relationship part in terms of qualitative as well as quantitative. But in today's talk, I'm just covering the quantitative structure-activity relationship, which is also called as QSR. If you see the European group, which is covered in this orchestra group, also called as orchestra group, or the REACH guidelines they follow to develop the mathematical model to show the interrelationship between the chemical compounds and their activity and their chemical properties. So if you see, they have mentioned 4 important points. I hope you will able to see it. First is to identification of the important compounds which have anti-cancer activity. Second is identification of the chemical properties which have high correlation, at least more than 60% correlation with the activity. It may be positive or it may be a negative correlation. And the third is validation. Validation should be there. And fourth is prediction power, how much the performance is of model is going on. So 4 major properties they have covered, and most of the European group of pharma industry follow these 4 guidelines of the REACH. This lacks last one principle of like OECD guideline compared to OECD guidelines. OECD guidelines, if you see the website and go to the tutorial part of the QSR model acceptance, or before like submitting your molecule for drug authority for approval, you should at least submit these 5 principles I mentioned here. If you go the tutorial, they have clearly mentioned what you have to submit the data before any approval. So they have clearly mentioned that only the in vitro activity is not sufficient. You should at least submit the scientific reason behind the activity. So you know, in vitro assay, there are like 2 types of the assay. We normally do with the cell-based assay and then go forward with target-based assay. Both are in vitro assay. And then after that, we go for in vivo study also. So if you see, the activity we find out normally, but at the same time, we do not know the answer, why the compound is so much active or so much less active. We don't know the answer by seeing the structure only. Because structurally, if you see, all the compounds follow the same elements. They have organic compounds, all organic compounds, you see, they have phenyl structure. Number of rings may be 2 or 3 or 4. But what is the like site of the activity? We don't know. What parameter should be there? We don't know actually. Even the compound itself not like able to say anything. It's you and your like study and your expertise required. A lot of like serious sitting on the bench with the chemist, with the biologist to find out the like relationship studies. If you see the prior study, first model like relationship is developed by Hans, and Hans is like called as father of like computer-aided drug design. And this equation, they relate the like biological activity with the log P value. What is log P value is? It is a lipophilicity measurement parameter which is responsible for biological activity as per Hen's law. So hence in 1969 they developed relationship as is shown here. They made like a correlation with the positive log P, which is the octanol water partition coefficient, which is mimicking the like absorption of the molecule from the intestinal membrane. So as you know, our bilipid layer is there in the membrane, is like made up of like bilipid layer membrane. So the molecule should have like lipophilicity as well as the hydrophilicity. Both the characteristics should be there for oral absorption of the molecule. So log P is a very important parameters to cover at. So this log P value basically most of the like time we analyze it, and there is one important rule called as Lipinski's rule of 5. I hope you heard about, especially the biochemistry or the medicinal chemistry of pharmaceutical students. So you just go through with the Lipinski's rule of 5. They clearly mention that log P should not be more than and equal to 5. Okay, so 5 is the cutoff. So while screening the molecules, we should take care of like to see the log P value of the molecules. If the log P value is quite higher than 5, then it's a violation of the like Lipinski's rule, and it shows that this molecule cannot be like a drug molecule. So we can like in the first step we can restrict the molecule. Similarly, there are like other rules, the other 3 rules, like the molecular weight should be like 500 or less than 500. Okay, and the hydrogen bond donor should be how much? Hydrogen bond donor should be 5 or less than 5. An acceptor should be, hydrogen bond acceptor should be 10 or less than 10. So you see the rule of 5 is indicating either the 5 value or multiple of 5 values. If you see the QSR approach, basically if you use the QSR approach at the first step of the lead screening, and in some stages we follow the screening process after the activity also. So it depends on the like project type and what the data you have in hand. If you are starting from the scratch, I suggest use the QSR at the first step, And if you have some in vitro activity data, you can use QSR approach in the middle also. So you see, like in the first step, you see the data set. If you like, data set is kind of like drug molecules in your hand with activity. You process the properties, and you find out the lead-based approach or target-based approach. Both are like very well like studied method. And first, like lead-based method is also called 2D QSR method, and structure-based or target-based method is called as 3D QSR method. You can use up to your expertise or lab expertise, and the service available with you. Any method you can use it. Both are very good method, but at the same time you should at least submit 2D QSR first. And after that identification of the lead molecule, we go forward with the designing of the molecules and synthesis of the molecule, in vitro activity and preclinical studies. And this way we can have a scientific report for step-by-step study of a molecules. This report actually required during the submission process. So I just cover some of the tutorial part also for the students' knowledge or information purpose. Because in QSR, the most important thing is to identify the chemical properties. So how you can like identify the chemical properties? Is there any like application or software you have? There are so many softwares actually. Commercially they are available, but you can use freeware also. Like Paddle is open source licensed softwares, you can use it. So you see in the first step, if you see the applicable domain, I mentioned it, is basically decided based on the structural properties and the relationship with the biological activity. In the process of relationship, there are a lot of methods you can use at the same time. I listed some of the methods here. For example, data pre-processing step, you can use normalization, standardization, feature selection, and outlier detection. Any one method or two methods you can use for your confidence level. And at the same time, for making the relationship between the biological activity and the compound, you should have at least relationship studies, what we call as multivariate analysis. So there are so many multivariate analysis methods. So in my lab, I use multiple linear regression, self-organizing map, principal compound analysis, you can use it, partial least square method, PLS method, and neural networks. There are so subvariants of the neural networks, and support vector machine. Even support vector regression method is also there. So you can check the validity of these models through some coefficient, statistical coefficient. Like R regression coefficient, R square is regression coefficient, R is correlation coefficient, Q square is cross validation coefficient square, and excuse me. Thank you. Another standard error also, you can like check the validity of your model. Similarly, you can like find out the properties through different methods. I just listed some of the descriptors. So then like if you see a lot of like descriptors are there for single molecules. Thousands of properties are there. So how you can like decide which one should be considered for your study? So there are like major 5 categories I listed out. Whenever you start with the relationships, try to select the properties comes under the constitutional descriptors, comes under the electronic and geometrical, hydrophobic, hydrophilic, and quantum chemical also. Even you can consider the topological. If the structurally they are very diverse geometry they have. So depending upon the like expertise, you can take the help of the chemist, sit with them, and find out the like which are the most important properties. This is a knowledge-based approach. At the same time, for if you do not have any knowledge, you can randomly use some standard softwares like Discovery Studio or Sibyl also there is, and Paddle also is there. And based on your like collinearity and other tests you can select randomly if you do not have a expertise. So use as per your expertise different method. And lastly, come to the prediction model. So once you develop the relationship, there is a mathematical equation which shows the linear relationship. This is called as multiple linear regression equation. And also at the same time, since it is covering the biological activity and the properties, so that's why it is called as QSR model equation. This equation basically used for further prediction of the molecules into active, inactive compounds. So once you develop the model with more than 60% accuracy, you can screen number of compounds, and this is a very fast method to screen out scientifically the important out of the outliers. So I just listed out the method of the QSR, so how you can follow in your lab. So first step you see, you have to select the experimental data in hand, divide into 80% into 20% ratio. 80% compounds with biological activity on the known drugs you can consider in the model development, and rest 20% you can use for the testing purpose. So that part is basically required for developing the model. And the second is, once you identify the molecules, you can predict the biological properties or chemical properties of the molecules, which is also called as descriptors identification or calculation process. And the third step, if you see, there is a relationship. You can use any method, as I mentioned earlier, any method. Regression is the best method for prediction modeling. And if you are looking for optimization, you can go for genetic algorithm also you can use. And for classification, you can use artificial neural network. And the last step is to make the set of rule to filter out the outliers. And this set of rule is called as, based on the QSR model, is called as applicability domain. So applicability domain is very much required for prediction of the random molecules activity, whether they fit in your model or not. So AP is basically required to filter out the outliers. You can define the molecular descriptor as represented here. These are the numerical values of the chemical properties which represent the chemical structure or the active compounds. So find out these properties as much as possible. It may be a 2D, mostly 2D properties, and some are 3D properties also there. So you can use any software to predict out, or any calculator you can use it, or you can take the help of the medicinal chemist also. So as I mentioned, 5 major categories are shown here. You can follow these categories. At least constitutional descriptor should be there in the relationship study. Electrostatic descriptor should be there. Topological and quantum chemical and geometry related descriptors. Try to cover all these 5 categories descriptors and make a relationship. I am sure you will get a more than 60% accuracy model. So I listed some of the software. Paddle is open source software. You can use this software in Linux as well as in Windows also in command line, and try to find out the results and see how much properties your molecule have. So I listed out some of the snapshot of the home page. You can check out 1D and 2D properties, 3D properties. And there are some library also, but I suggest don't take libraries because they are fragments. And fragment basically they have considered as a pharmacophore. So I don't think pharmacophore consideration in a property is a good enough approach. So avoid that component. Other open source software you can use in Linux is Moderate tool, which is also available. Go to the GitHub and download it and try to install it in your system and try to calculate the chemical properties of the molecule. It also covers a lot of properties. And rest of the softwares are very commercially available and they have a very high price actually. So what is the step-by-step approach to develop a model and virtual screening of the anti-cancer agents? So I listed out some of the steps as I mentioned earlier, that to select the training set, then to calculate the properties, make a relationship, filter out the outliers, make a applicable domain rule, and then predict the properties of the molecule. Random molecules. Based on the prediction, you can screen out outliers and only suggest few molecules for further testing. This way, you can save the wet lab economy. At the same time, you save the life of the small animal. So this is the alternative method in compared to in vitro assay, so that you can prioritize the molecules and save the life of the small animal. So this is a flow chart of the QSR method which shows that training set compound should follow these models. It may be a regression or classification method or matrix-based method. It's depend upon like expertise you have. If you have a matrix-based approach, you can use PCA method for that, and make a prediction of the molecules of untested molecules randomly. Before in vitro study, you can get an idea that how much of if you have 100 compounds, you will get an idea that out of 100 how many compounds are good enough for further activity evaluation in lab, in vitro lab especially. So I listed out this is a snapshot of the some of the properties of the molecules. You see each column represent each chemical properties in terms of numerical values. So they are dependent on the each row indicates the separate compound. All compounds are anti-cancer compounds. Each column represent different properties, and you have to find out 4 or 5 properties which are most related with the activity. So how you find out this property? You make a correlation. The correlation matrix is very common method, a very easy method. Otherwise, you can use PCA also, principal compound analysis. In this PCA, I just shows you that you have multiple dimensions, you can reduce the dimension up to 2 or 3 properties. So I mentioned that PC1 and PC2 is out to 5 columns, you have to ultimately get the 2 columns. Only 2 properties are basically related with the activity. So this method is basically uses the covariance matrix preparation and then eigenvalues and eigenvector determination. Based on that, you can predict the important chemical features. So PCA at the same time used by different scientists and for different purpose. I mentioned some of the application to identify the pattern in the data, to detect the correlation between variable. Even in HPLC and GCMS, you can use the PCA. To reduce the dimensionality, if you have a large enough data and you are not able to get the idea where to start, you can reduce the data through PCA. To find out the important features. At the same time, in chemical, in our cases, skin QSR, we use this method to find out the chemical features. So I listed out the result of the PCA. As you see, there are so many PC dimension components, PC1, PC2, PC3 in each different column. And in different rows, they have different compounds actually. So you will get most important component, most important dimension through PC analysis by plotting it. You see the plot out of PC6, we only selected PC1 and PC2 based on the high coefficient values or the PC component values. So this way you can prioritize the in terms of percentage as you'll plot it in graph to show you which properties have a maximum effect on the chemical compounds. So the first property is indicating high rise bar, which indicates very high correlation with the biological activity. And at the same time, you will see second property, second column. You can see third one is very less actual compared to PC2, PC1. Third and fourth. Only 4 properties basically shows high correlation with the biological activity out of 1000. So you see this is very, very fast and efficient method with scientific reason. It correlates the activity with the biological compounds or drug compounds. So what are the advantages of the QSR? As I mentioned, it is very fast method to identify the important lead compound out of the so many virtually designed or so many synthesized molecules. You can select very limited set of compounds through this scientific method. It requires knowledge of the little bit chemistry as well as some of the statistical approaches or machine learning methods. If you have large enough data, like you have more than 1,000, 2,000 compound, you can use the machine learning method like artificial neural network or SVM. Otherwise, if you have a less data, you can use a statistical method, PLS or regression, MLR, and so on. So it helps, ultimately model helps to predict the active, inactive compounds. At the same time, it prioritizes the molecules. And at the same time, it saves the wet lab economy. So unnecessary like synthesis of the compounds, you can stop through this method. Unnecessary testing of the molecule, you can stop through this method. And it helps the chemist a lot to find out the potential lead. Otherwise, earlier method is structure-activity relationship, SAR analysis they do, which require knowledge of the like pharmacophore, and they do change in the site change, and then predict then they evaluate the activity through in vitro assay, and number of times they repeat the same thing with different modification at the side chain around the pharmacophore. So pharmacophore considered as a conserved active part of the molecules. But where is it? We don't know. There is no specific rule. Only chemist knows that. Where is the like pharmacophore? So to overcome this problem, this approach QSR basically helps a lot to the chemists, because chemists are unable to explain why this pharmacophore region or the fragment part conserved in nature have an activity. They don't know the reason why it is so much active. So how to like answer this question? You can answer this question through QSR approach only. At the same time, there are a lot of advantages, but at the same time, there are a lot of disadvantages also. If you do not have a sufficient knowledge and you do not have an expertise, or randomly you are trying to develop a model, you make a very fake model. It will fail, actually. So like where are the points where we fail at the same time? So one is like, mostly if I see in a new generation research paper, I seen they lack make a relationship study. They lack a good relationship in the model. And they make high assumption and hypothesis based on the statistical parameters only. That is not a good approach actually. Whenever you like try to make a relationship, at least see the validation part. I'm showing you some of the snapshot where you like find out where is the error is there. Although all the parameters looks very good, but at the same time, if you see the validation part, you will recognize that there is a mismatch actually. Some error is there. So as I mentioned, application of QSR, I just show you like biological activity prediction of the chemical toxicity, if you are using the like toxicity part, and at the same time molecular properties prediction, what we call QSPR, if you are taking mice or rat. So for a single test, it required almost 10 mice or 8 mice for a single set. So it repeated 3 times. You can imagine like 30 mice required for detection of LD50 only. So 50% mice you have to kill. And then you get the value of LD50. So what is the alternative method? So this is the alternative method, and most of the developed country using it. In India, we are also using it in our research lab. Now let's see where the machine learning methods where we can use in this approach. So you see, I just mentioned some diagrammatic way. What is machine learning? Any idea? If you are using like any method in computers and enable the computer to do something with your training, this is called as machine learning method without any programming. For example, you are like mostly doing it in Excel, you develop a formula and predict the values by copy paste. So that equation formula basically given within the MS Excel, that again are like machine learning predictions. Okay, without any programming, you done the like sum, division, function, all sort of things you can do in Excel. Mostly we use it very commonly. It is a machine learning approach actually. Similarly, you can develop number of like relationship model, and these models are mathematical nature, so they require some formula and you can predict the values. So our objective in this study is to predict the biological activity. Either it may be IC50 value, or it may be a Ki value, inhibition constant, or dissociation constant, KD values. So Ki and Kd values basically required when you have like going for in vitro target based assay. Like you are like finding the concentration of the molecules on the target. It may be in the terms of like micromolar and nanomolar. So how you like predict this concentration at micro level or nano level? You can use the QSR method and develop your own model. So whenever you start with the training, as I showed in the flow chart, earlier the program follow the data, and at the same time you have to put the program, some script. And it give as an input, it give like after programming, give some output. That is a traditional approach, like in C and C++ program, earlier we did in our graduation or post-graduation level. But nowadays, there is a different approach. If you see a lot of programs already written, you have to just copy and paste it, and it will give you the results. Here we are giving the input data set like experimental activity and molecules with their properties. At least two variables should be there. Before any relationship, you should have at least independent and dependent variables. So here independent variables are descriptors or chemical properties, and dependent variable is biological activity. And you are trying to find out the relationship. So whenever you use the like independent or dependent variable, there are like mostly two approaches used, like correlation and regression. But there is a relationship, there is a difference between correlation and regression. Regression is always used for prediction model, differentiating the model in terms of predictions. Correlation make the correlation only. It may be a positive correlation or negative correlation. I hope you heard about in your study. So you see, we put the input value, which is a biological activity and the chemical descriptors, and at the same time we are like we are giving the output also. Output is the biological activity actually, because we know that like this is a known data set. This is an experimental data set where we know the compound and their activity. So we are giving the input data set compound with their activity output, and at the same time we are using the machine learning method which is already available, which is free of cost. You can use it. Based on your knowledge, you can use any method, but you should have at least some idea which method we can use. So you should at least discuss with your teacher and select appropriate method. I am sure you will get, without any programming, you will get the result. You develop the model actually. Model develop. Once the model develop, you can use this model for prediction of any number of compounds. You can predict any biological activity. It may be IC50 or Ki value or GI growth inhibition factors. Depend on the like experimental project, you can develop any model. So what are the like different method in machine learning? You see, whenever we give the data, whenever we know that like there is a experimental data, because most of the time the research experimental done for number of compounds, and it suppress in the form of like file or project reports. That's it. But we do not know like how to like save this molecules, how to use this biological activity for making the model. So we are using the prior study like 30-40 years data and make a relationship. So and developing a model. So this approach is called a supervised approach. There is a supervision actually. Experimentally labeled data if you have in hand. So whenever a labeled data is there, it is called a supervised learning. If you do not have any prior studies data and randomly starting with predictions, making use of some standard formula or some kind of program, that area is basically comes under artificial intelligence or machine learning, and this method is called as unsupervised method. For example, like searching you see the like searching in Amazon or Flipkart. So whatever you put the information, next time whenever you open the Flipkart or Amazon, you see likewise your likelihood based prediction they made at the same time on the screen. So how the program itself learn it, based on your interest, they find the appropriate item and showed you in the screen. Each time whenever you select the compound of your interest, they know the like software itself trained itself that the requirement of the molecule or requirement of the item is there. So this method is called as unsupervised method. So I listed some of the like method. Reinforcement, which is a reward-based approach. Mostly we do not use this one because it gives a lot of like error. And the second is, which is most successfully used in lab, is supervised method, which is also called as inductive method. And the third is semi-supervised method, which is also called very effective method in supervised learning. And the last is unsupervised method. So sometimes you get the data in lab where you do not have any information, any background information or any prior information. So you can use unsupervised method. I suggest you some of the method I listed, like clustering, you can use. It is very successful and gives 90%, 80% accuracy. And in supervised, you can use regression or classification method, any method you have. So in regression, there are like linear, simple linear, multiple linear, and non-linear methods are there. And classification, you can use up to the, like, based on your expertise, you can use decision trees, Bayesian analysis, and logistic regression, random forest method, even the ANN and SVM also. So what are the steps of machine learning? Whenever you go with the model development, you should follow this standard approach. Make use of the data collection, data pre-processing, then model selection, model training, testing, and tuning. Model tuning is called as optimization also. And the last is model predictions. Once you get the model for a chemical series, you can use any number of times. So how to make use of the training or data set or supervised training data set? So you see, I just listed out that whatever the experimental data you have in hand, use at least 80% in the training part, as I showed in the upper region in the diagram, and the lower part, like 20%, use for testing purpose. So it is a like unbiased modeling approach. So it gives you a good relationship. X and Y's are the like independent and dependent variables as shown here. So you can use independent variables number of times. There is no limit, but dependent variables should be one only. It may be a biological activity, for example, in vitro assay, for example. Similarly, after making a relationship model, you can use some of the component of the machine learning for validation purpose. I listed some of the steps of modeling. I'm not going in detail, but listed some of the like areas where you have to submit, which require during the submission of the like proposal or any kind of the research paper in journals. They require representation data, they require evaluation data, they require optimization data. So you can give any one approach at the same time. I listed some of the here, but you can use any one method. Similarly, types of learning, you see, there are so many learning types as mentioned: supervised, unsupervised, semi-supervised, or reinforcements. So you can use any method based on your learning and ability or the expertise available in the lab or in the group, and use this method, any one method, and try to develop the model. Now I just show you like the example of the like already done exercise. So these are the drugs, known drugs, which are derived through QSR models, and you're using in your daily life actually, like metconazole, lomrazine. So they are all developed through QSR approach. So it's a very fast and aggressive method. You see in this diagram shows that the upper part is the training data set with LD50 value, which is in normalized form. And then we made the relationship model with the regression coefficient of 99% and cross-validation regression coefficient is 96%. You see the linear line is there within the graph. It shows that there is a linear relationship. And you see in the below part, there is a QSR equation which shows the important chemical 5 properties out of the like 1000 properties, which having a relationship with the immunomodulatory anti-inflammatory activity. So this model is developed for gallimonic acid, which is a natural compound, which shows high anti-cancer activity also. So this is a published data. I just taken out the snapshot of my own research work. You see gallimonic acid, number of derivatives below part you see in the table. Number of designed molecules G4, G5, G6, number of designed molecules they design, And with the help of this model, we screen number of compounds. We made the prediction of the activity and select the best one. So lower the concentration predicted value, higher the activity. This is the formula. So we select the lowest concentration and predicted log LD50 and later on done the in vitro and in vivo activity, and we found that prediction is good enough actually. This diagram shows that how to make the model and how to check whether the model is good enough or bad. So as I mentioned earlier that this model is basically based on OEC guidelines. So there are different parameters I showed you. So for explanation, I will take more time actually. So I'm just focusing on that part where you have to see whether the model is good enough or not bad enough. You see the last two graphs, training set mapping graph and test set mapping graph. They are overlapping with each other. Blue line and red lines are overlapping with each others. So this is a validation step where we see the prediction of the molecules with the experimental values. So if the experimental data and the predicted data overlap with each other with 90% or more than 90% accuracy, then it means the model is good enough. So you see in the test part and the training set part, both are overlapping with each other. It means this model is good enough. This already model published in POS 1. I just taken some of the results. And you see this, in this model, you see both the diagram. The red lines and blue lines both are not overlapping with each others. Okay. Red lines, and if you see the training set data, they are slightly overlapping, but you see the test data set, second diagram, test data set lines, they are not overlapping with each other. Although the correlation coefficient is 0.85, you see 85% is correlation is there. And prediction accuracy, which is represented by QSCAR, which is 79%, which is good enough actually. The minimum selection is 60% actual. Cutoff is 60%. Still, the test set data set shows that there is an overlap. So it's a bad model. It will not accept it actually. So we normally reject this kind of the model for virtual screening. You can also check your model with this approach. This is another example where we developed oxalic acid model, QSR model for prediction of anti-cancer activity, and later on tested in vitro and synthesized the molecules in lab. And we find out that there is a good two cancer cell line model we have developed. One is lung cancer cell line and CNS related cancer line with good correlation and good cross-validation coefficient. More than 85%, 80%, and 98%, 96%. These compounds of the ursolite, which is a natural triterpenoid compound, these compounds are very well used in anti-cancer activity related research work. Even you can use for in your lab also. These are available with the pharmaceutical companies. So this work is also published in JMM. You can get more detail of this research work through this journal. This is the example of machine learning approach where we use the clustering approach. When, as I mentioned, when we have a large data set and all compounds have diverse structural formation, you can classify these compounds into different clusters. As you see, out of you see 2,291 compounds, all are anti-breast cancer cell line active compounds, anti-cancer compounds. But if you see the structure of these 2,291 compounds, you will surprise that they are not similar in nature. Structurally, they are different in natures. I divided these 2,000 compounds into 8 clusters based on their structural similarity. So we made it 8 clusters. Each cluster represent almost, you see in the first column I mentioned the number 61 by 5, 643 by 5. And except cluster number 6, rest of the clusters have sufficient number of compounds. So how much compound should be there in the training set? The threshold should be 30. at least you should cover 30 compounds to make a relationship model with known activity. So accept sixth model, rest models basically, and develop a relationship model, what we call the support vector regression coefficient, and we predict the anti-cancer compounds relationship in eight clusters. Each cluster represents different centroid molecules, as shown here. Cluster 1 is all are structurally diverse, but the beauty of these compounds are they are known anti-cancer agents. You see, structural wise they are different, but activity wise they are similar. So why there is a similarity in these activities? So to answer these, we made 8 clusters because these 8 clusters have different properties with their activities. And this method is basically used to screen out random compound. We are using in CMAP to screen out any compounds. So once we, like you see on the top, there is a small molecule I mentioned here, and randomly taken it. And if we want to like predict the biological activity or anti-cancer activity against breast cancer cell line, we just try to match the cluster first. So first step is to match the compound with the cluster, in which cluster it lies, in which clusters structural similarity is there. So out of 8 cluster, only 7 number cluster shows the similarity. This molecule is basically videnolide derivative. So I taken it from Videnia, taken it from the videnolide molecule for cluster number 7 and predict the anti-cancer activity. And at the lab level, we find out that the same activity we predicted, we get in the in vitro test activity also. So this way we can screen number of compounds for anti-breast cancer cell lines. So we have made almost 20-30 models we have in CMAP for virtual screening for different chemical series, especially the triterpenoids, diterpenes, flavonones, dietary monoterpenes, and recently curcumin derivatives. So these are like models we have in CMAP. We use for screening purpose. You can also develop as per your expertise, because in CMAP there is we have a mandate to follow only the natural compounds, but you are free to do anything. So you can develop any model for any compounds. After like getting the prediction, we check the ADME part through ADME rule, and the molecules comes under the eclipse that shows that the ADME is good enough. We select that molecule for further activity, in vivo activity. And you see this table shows that predicted toxicity we have mentioned here for cytotoxicity, mutagenicity, skin irritation, and tumorigenicity. So these are the xanthone derivative I mentioned here. And you see different at high level doses and high long-term use, they show the toxicity in human. So red uncircled part indicating that you have to see these areas whenever you analyze the molecules. This diagram shows that after like QSR, you just try to see the binding affinity of your molecule with the target. So you can do this exercise through molecular docking. So first diagram shows the docking results, and the second part shows the pharmacophore mapping regions. So if you have expertise, you can check whether the molecule is good and have space to bind on the target conformation or the binding pocket. You can download the crystallographic data, protein crystallographic 3D structure data from the PDB, and see the binding affinity on that specific pocket. This area is called as pharmacophore modeling, and check whether the molecule's size is good enough to bind there or not. And later on, then you can also same time predict the metabolite synthesis. After docking, you can see the, you can do the one more thing. You see the binding stability of the molecules through MD simulation. You can run the like video. There is a last one. Through this molecule, you can like visualize the molecule, small molecules binding on the protein. This is a small molecule which we have taken here, is basically marinol molecule, which is a flavonoid. In the diagram below, there is a play part. Click on that. Okay, anyway, just leave it. Anyway, leave it. Next. So whenever you do the MT simulation, you can check the binding stability up to 30 nanosecond or 10 nanosecond. It depends on the hardware you have in hand, and you see the stability regions. You see the last diagram at the 4000 picosecond, molecules break from the binding proteins. So we can reject these molecules for further testing. It will not be good molecules. Rest of the peaks shows parallel. Red peaks indicates the experimental part, positive control, and negative control is represented by black color. So this way we can screen the molecules for validation. Next. So this is the overall view of the QSR. I hope you got the idea how to screen the molecules, large enough molecules, and use the QSR approach for small molecules. I don't recommend for large molecules or protein level study. Small molecules is good enough approach. If you have any question, you can ask me. Thank you so much.
Thank you, sir, for such an engaging talk. The session is now open for question and answer.
Yeah, students from you.
Anyone? Okay. I'm
here for you only. I'm here just for you only. So ask the question. So do you know the difference between regression and correlation? Learn it. There is a huge difference actually. And how to find out the data? How to decide whether there is a positive correlation, negative correlation? And which data is good enough for regression analysis? Try to learn its homework. Okay, thank you.
Thank you, sir. I would now like to request Professor Khan to kindly present Dr. Khan with the memento. Thank you, sir. With this, we conclude the current session, and I would like to invite all the guests and participants for high tea. Thank you.
To the students, just now, Professor Dr. Khan has very rightly put a question, correlation, regression and all that. To be very frank, did you come out in your learning at any time? At any time? Okay, this made me realize of 1982 when I joined for MSc at Pantnagar in the course or to be offered the titles. Then we had one elementary mathematics. हम लोग सब बायोलॉजी वाले क्यों भागते हैं? मैथमेटिक्स छोड़ के भागते हैं, टू बी फ्रैंक. Okay, you know, in that we were taught what is trigonometry, calculus, differential integration. Okay, then complete. Okay, this is the way how to learn. Then it will be very easy to understand what the module has been sent, discussed over here. Okay, that means what I draw the conclusion, you all must be saying, that is before taking any molecule for study, and the molecule, first you got to do, okay, simulation and all that, and then go and rightly pointed out. None of you asked why only small, why not big? हाँ, छोटा molecule लेना, बड़ा क्यों नहीं लेना? है ना? Yeah, I was just going. He was insisting on that. Again, pointed out not the bigger one, the smaller one. है ना? तो आप लोग है ना, when you're attending and all that, we should be very cautious what each one is being told. Okay? ये ज़रूरी नहीं रहता कि सब कुछ हमको पता है या उसको नहीं आता. If it is not known to you, you should have that much of intuition in you, okay, to ask. So that is a process called as a learning. Okay. Now this is a platform, very rightly says, because I'm on the second day today, right, about 2-3 talks and all that. So much informative, okay, presentations have been done. So one should take advantage of that. Thank you, Dr. Khan. You have rightly presented, okay, but I'm not disappointing you all or something, okay. Yes, I want to say, जरा इंटरैक्टिव होना चाहिए, ठीक है? ऐसा है ना, ऐसा समझते हैं, ऐसा हो जाए तो समझ जाए कि सब कुछ समझ में आ गया, या नहीं तो दूसरा है कि कुछ नहीं समझ में आया, है ना? दो ही होता है ना? तो ये हैसल से आप ऊपर आइए जब भी लर्निंग प्रोसेस कंप्लीट होगा, यही तो आप ऐसे रहेंगे तो नहीं चलेगा, है ना? I don't know how many of you after the classes and all that ग्रुप डिस्कशंस वगैरह करते हैं आप लोग? नहीं, ग्रुप डिस्कशन वगैरह कुछ होता है या नहीं आप लोगों के बीच में, है ना? हाँ, अरे ग्रुप डिस्कशन किसका, सब्जेक्ट का? हाँ, जो पढ़े हैं उसके लेकर के, है ना? Whenever come across the students, I want that, because I recall of 1982 to 84, जब हमारे पास एग्जामिनेशन होते थे तो we are only admission was only 5 seats. Till today it is 5 seats. Okay. Then what happened? Before the examination, we used to get in the hostel. Preparation कर रहे भाई, मतलब हमको 10 बजे चाय मिलती थी. तो हम 4-5 लोग टॉपिक डिसाइड किया, डिस्कशन रहेगा, ठीक है? चाय पीने के बाद 10-15 मिनट जुड़ गया. Then each one of us used to boost the questions, है ना? टारगेट करते थे, आप बोलो, डिस्कशन होता था. Most of the things were clear, and today also sometimes one of my batchmates, Professor Zargar from Kashmir University, okay, Sher Kashmir, when we okay met, तो हम लोगों ने अपना ज़माना याद किया. अरे याद है हम लोग कैसे खिंचाई करते थे, है ना? ऐसा क्वेश्चन पूछते थे. That was the best way. That देखिए, group discussion, don't take it is only for the management courses. Group discussion हर सब्जेक्ट में आ सकता है, provided one have that intuition. Okay. So with this comment I had, not to take a lot of your time, बोल तो बड़ी informative and concluding बोल तो लिखिए आप लोग ना कि way of presentation. That's why he was insisting. Then he pointed out for the students, कह रहे हैं. I'm not disappointed. मतलब आप लोग है ना अपने आप को, है ना, एनर्जाइज करिए इसमें. कभी भी है ना, कोई भी लेक्चर वगैरह कोई क्लासेस में भी, ओके, क्लासेस में भी हम कहते हैं, क्लासेस में भी हो रहा है, पढ़ रहे हैं तो 50 स्टूडेंट है, पढ़ रहे 50 समझ में नहीं आया, अरे पढ़िए, पूछेंगे तो समझेंगे कि हमको कुछ नहीं आता. अरे भाई सबको सब एक ही नाव में तैर रहे हैं, कोई पूछता नहीं, यही रहता है फीलिंग. इमीडिएटली बी फ्रैंक टू योर सेल्फ, रेज द हैंड and ask the question clear, है ना? नहीं तो क्या होता है, मेकिंग ए माउंटेन ऑफ ए मोल, है ना, वो बढ़ते जाएंगे आपके सेल्फ का. फिर उसके बाद क्या होता है मालूम? व्हाट माय लास्ट फ्यू मिनट्स से लोग फिर क्या करते हैं, मगिंग, रटना, इतना ऐसा 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 ऐसा. अब वही आदमी जब टीचर बनेगा, मगिंग काम नहीं करेंगे, समझना पड़ेगा उसको और पढ़ाना पड़ेगा, क्योंकि जब स्टूडेंट पूछेगा तो बताना पड़ेगा, है ना? अदर साइड, यदि कोई स्टूडेंट टीचर को पूछते हैं सर व्हाट मैडम व्हाट इज, बोलता यदि सही टीचर है उसको पता हो तो बता देगा, नहीं हो तो, है ना, बोलेगा आई विल जस्ट रीड एंड नेक्स्ट क्लास आई विल क्लियर दैट. नहीं तो फिर क्या करता टीचर, इतना आसान, यू डोंट नो, आई डोंट नो हाउ केम टू दिस क्लास, आई डोंट अंडरस्टैंड. हाँ, ये रियलिटी बता रहा हूँ मैं. ये टीचर की क्वालिटी है. टीचर का मतलब है डेफिनेशन टीच करो, है ना, उसे भागो मत. Some of students, इसीलिए मैं कहता हूँ, I define teacher is a student first. Teacher पढ़ा रहा है, मैं तुझे. To me, the people, students ask the question. If I know, I immediately clear. नहीं होता तो sorry, next class. So for myself, I go, take the question, read and all that. Next class, I start the lecture. First, I will answer that question and then proceed. This is the method of learning होना चाहिए. Teach and thought जो method बोलते हैं ना, ये होना चाहिए. तो ऐसा है ना कि I congratulate, excellent lecture was there. Man, he was insisting for the students, है ना. मगर ऐसा मत करो, हाँ. जरा एक्टिव होना चाहिए, है ना. क्योंकि it is a very challenging world. ठीक है. इसको इस प्रोफेशन में कंटिन्यू करना है तो आपको है ना मेहनत करना पड़ेगा. You cannot be just a very good listener, है ना. उसके अलावा भी कुछ है ना. So this is all my suggestion. I'm not discouraging this, just my advice. ये मेरा काम रहता, कभी उसको इनको पूछ लो, आपके ताबीज़ वगैरह किसी को पूछ लो, they will all students, है ना. We used to interact and बहुत बार आते थे. जब I know him since 94. Okay, was my student at Faizabad. So पूछिए, तो सुबह फ़ोन नहीं करना, सर आप तो आ रहे हैं ना. मैंने बिल्कुल आ रहा हूँ भाई, ऐसी बात नहीं, आ रहा हूँ मैंने. ठीक है. यदि आप 24th का बोलते तो हम नहीं आ सकते थे, क्योंकि I had some meeting, official meeting, 23rd and 24th. So this is the way. Okay, all the best in the next sessions. Also be interactive. Okay, that's all.
Thank you.
Thank you.
Thank you everyone for joining us back for the session 2. In the session 2, I would like to introduce Professor Nikhit Jamal Siddiqui and the Dr. Sarika Singh. Professor Nikhit Jamal Siddiqui, she is a professor in King Saud University, Saudi Arabia. She has completed PhD in biochemistry from Dr. RML Awadh University, Faizabad, India. Her expertise includes biochemical toxicology, collagen, hydroxyproline, biochemistry, and nanobiochemistry. She is a member of British Society for Parasitology, Indian Society for Parasitology, Indian Society of Biological Chemistry. She has been teaching on undergraduate and postgraduate biochemistry students. Thank you, Professor Nikhit Jamal Siddiqui. Now, I'm going to introduce our next chairperson, Dr. Sarika Singh. She is a principal scientist in CSIR CDRI. She completed her PhD in biochemistry from Lucknow University. She did her postdoc from the UT Southwestern Medical Center, USA. She received the Raman Research Fellowship, CSI India. She is the awardee of Indo-US Fellowship. She is the team member of CSI Technology Award. She published various research papers in various reputed journals. She has been working with in CDRI since 2006. Now I would request Dr. Khan, please present memento to the Dr. Sarika. Thank you, ma'am.
Moving on to the first talk of this session. So to know more about advancement in cancer, we would like to call Dr. Rohit Saluja, sir, to share some knowledge on cancer. He is currently working as assistant professor at AIIMS, Hyderabad. He has been awarded Ramalinga Swamy Re-Entry Fellowship 2013 to 2014, DBT India, and Travel Grant Award for 9th EAACI GA2 LEN Winter School, Basic Immunology, Research and Allergy, and Clinical Immunology in Davos, Switzerland. His current research areas include biochemistry, immunology, mast cell biology, allergy, and asthma immunology. His research projects include clinical and molecular study to explore the effect of homeopathy in psoriasis, role of pectin in prevention of food allergy, etc. So I would request Dr. Rohit Suluja sir to present. Rohit sir, please open the camera and unmute yourself.
Yes, sir, your slide is visible.
Thanks to Dr. Tabir for giving the opportunity to discuss the work, and also all the organizers who are putting the constantly effort to make this conference successful. So today I'm going to talk about the role of interleukin 33 in inflammation and cancer. So if you talk about the allergy and inflammation, then the first cells comes into our mind is the mast cells. Other than the mast cells, basically the eosinophils and the basophils are also play important role in the allergy and inflammation. So why mast cells are important? So because the mast cells are the key player cells in the allergy, because they express the high expression of the receptor. It's called the FcFnR1, and this receptor is responsible for binding of the IgE, and this IgE can activate the mast cells, and then mast cells can degranulate and release their mediators. So as you know, like the histamine is the one of the most important mediators released by the mast cells. So whenever you have the allergy, you go to doctor, and then doctor, I think 99% they will recommend you the antihistamine. So basically they are involved in the pathophysiology of the allergic disease such as rhinitis and asthma. So why mast cells are unique? So mast cells are unique because of the two features. One is that they can live for the long time. So if you have seen your CBC, that is called the complete blood count, then you can see that you have the different type of the blood cells in your body, like neutrophils, PBMCs, eosinophils, basophils, and many other cells. But you won't find the mast cells there. Why? Because mast cells are not present in the blood. They are present in a negligible amount that you cannot detect it. But they are mainly localized in the tissues, like the skin, GIT, lungs, and this is basically make them statistically significant for the first lines of the defense. Because we are here in the external environment, and there are a lot of pathogens surrounding us, but still we are healthy. Why? Because we have the mast cells in the skin, and that mast cells are basically fighting 24/7 against the pathogen. Simply when we eat the food, so we not only eat the food, along with the food, we take the pathogen, especially if we are taking the Golgappa and this type of the thing. So, but thanks to the mast cells, that you can eat the Golgappas because mast cells are located in the GIT. Similarly, when you inhale the air, then you not only inhale the air, you also inhale the pathogens. But thanks to the mast cells, they are present in the lungs. So basically, once the pathogen reaches to the lungs, they can fight against the pathogens. So basically, mast cells can act as a first line of the defense against the invading pathogens. If you see the reactivity of the mast cells, so mast cells can express different type of the receptor, as you can see it here. One of the most important receptors is called the Fc epsilon R1. This receptor is basically responsible for binding of the IgE. Similarly, this receptor is responsible for binding of the stem cell factor. So stem cell factor basically, So stem cell factor is basically responsible for mast cell proliferation, and the recent is called the ST2, and this receptor cells and how the signaling pathways can happen. So if you have to remember that IL-33 is the ligand and ST2 is the receptor. When there is a ligand receptor interaction, then there could be the activation of the cells, and cells can release different type of the mediators, right? So here you can see that the one mast cell get activated by different stimuli, they can release different type of the mediator. I think one of the mediators that you already know, and that is the histamine, most important mediator from the mast cells. Other than the histamine, they can also release different types of the prostaglandins, cytokines, chemokines, leukotrienes, and all these mediators basically play important role in the allergy and inflammation. Here you can see the animation that how mast cells get activated. So once a particular ligand binds to the mast cells, then you can see the activation of the mast cells. Right here, you can see these granules are reaching towards the surface of the cells, and then they can get activated. They release their mediators. Like histamine is the one of the most important mediators released by the mast cells. Similarly, they can also release a secondary mediator like the phospholipid-derived arachidonic acid or the platelet activating factor, tertiary factors like the de novo synthesis. So what are the stimuli required for the mast cell proliferation? Because the proliferation is one of the most important phenomena in the body. And so as we discussed that the stem cell factor, basically it is required for mast cell proliferation, and for binding of the stem cell factor, we need the receptor called c-KIT. Why we need the mast cell proliferation? To maintain the homeostasis. If you don't have the mast cells, then you will be the happiest person, right? But it's not right. Why? Because you could be happy because you don't have the allergy, but at the same time you will be immunocompromised, right? Because mast cells will not be available to fight against the pathogens. So whenever you are in the external environment, your body will not be able to fight against pathogens, and then you will be sick. So basically, it is important for the maintaining the homeostasis. If the mast cell number get increased, then there could be a disease called mastocytosis. If there is a less number of the mast cells, then it will be like immunocompromised. So basically, interleukin 33 that has been described to be upregulated in the allergic condition. So we hypothesize that IL-33 can also induce the mast cell proliferation. So we tested it in the in vitro as well as in the in vivo condition. So now I am introducing the new terminology, that is the interleukin 33. So you may have heard about the different type of the cytokine like interleukin 1, interleukin 2, interleukin 6, interleukin 10. All the cytokines are well described in your textbook, but you won't get the information about the IL-33 because this is the recently discovered cytokine. It's a very young cytokine, I think hardly 15-year-old. So it belongs to the IL-1 superfamily, and IL-33 can induce different type of the immune cells such as the mast cells, eosinophils, and the basophils. When these cells get activated by interleukin 33, they release the type 2 cytokines. What are the type 2 cytokines? Like IL-4, IL-5, and IL-13. So as you know that the ST2 receptor is basically responsible for binding of the interleukin 33. When interleukin 33 binds to the receptor called ST2, this receptor forms the heterodimer with the IL-1RAP, that is called the IL-1 receptor accessory protein. So there is one receptor, there is another receptor. So basically it is called the heterodimer. So this heterodimer is responsible for binding with the interleukin 33. If we see the signaling pathway, then basically it is the NF-kappa B and MAP kinase pathway that is governed by IL-33. Here you can see the signaling pathway. So here is the IL-33. Now we know the name of the receptor, right? It is called the ST2. This receptor forms the heterodimer IL-1RAP. Then there is a downstream signaling pathway, MyD88 adapter protein, and then further downstream you can see the NF-kappa B or MAP kinase pathway that can release different type of the cytokines and the chemokines. They play important role in allergy and inflammation. So now coming to the IL-33 basic biology. So now the first question is if IL-33 is constantly released or it need any stimuli. So the answer is it needs the stimuli. It is basically present in the nucleus of the cells, but when the cells go for the necrosis, which type of the cells? Basically it is a structural type of the cells like epithelial cells, endothelial cells, smooth muscle cells, fibroblasts. So when these cells go for the necrosis, then IL-33 comes out from there, then it can be recognized by the ST2 receptors, and then further downstream, NF-kappa B pathway, or it could be the MAP kinase pathway that leads to the cell differentiation and activation. Now coming to the IL-33 induced signaling pathways. So as per our basic knowledge, we know that stem cell factor is binding to the c-KIT, and this binding leads to the proliferation. So our query is whether IL-33 can also induce mast cell proliferation or not, right? So the objective is to investigate the effect of IL-33 on the proliferation of the mast cells and explore the signaling pathways that is required for this process. So for this study, we injected the interleukin-33 in the mice, in the peritoneum as well as in the ear for continuously 3 days. On day 4, we collected the peritoneal lavage and then checked the number of the mast cells as well as the Ki-67 expression. So Ki-67 is basically the marker for the proliferation. In the ear of the mice, we found that IL-33 is basically responsible for high number of the mast cells as compared to the vehicle, and then you can see a significant difference there, right? So can you see it is a real proliferation, or it is only the increase in the number? The possibility could be like the mast cells are migrating from one place to another place, and then they are basically increasing in the number rather than it's a real proliferation. Right. So what we have done, we have looked for the proliferation for checking the Ki-67 expression by the confocal microscopy. So here you can see this blue color is basically showing the TAPI. It's indicating the nucleus. Red color is showing the evident TR. It's showing the mast cells. The green color is the Ki-67 expression, showing the proliferation marker. And the last picture is basically the margin. So here you can see there are many cells. These are the nucleated cells. There are many mast cells also there, and these mast cells express the Ki-67, means these mast cells are basically proliferating. But if you see the vehicle, so there are the, yes, we have some nucleated cells, we have some mast cells, but if you see in the green color, we hardly have anything. So green color is basically indicating the proliferation. That means you hardly see any proliferation in the vehicle. Similarly, we have seen in the emerging stage. So basically, this experiment indicates that mast cells is basically getting proliferated after the stimulation with IL-33. When we injected the IL-33 in the peritoneum, we get the more number of the mast cells as compared to the vehicle treatment. And again, we check the expression by Ki-67 in the flow cytometry. Here you can see there is a significant peak here that is absent here, right? So basically, it indicates that IL-33 is responsible for at high expression of the Ki-67. Overall, this result indicates that IL-33 is responsible for mast cell proliferation. So this is basically the in vivo result. We confirm the same result with the in vitro by using the bone marrow cells that were further differentiated with the interleukin-3, and then we get the bone marrow derived mast cells. And as we discussed that the mast cells has a unique property for having the high light response, means it can survive for the year. So if there is any contamination in the bone marrow cells, because bone marrow cells have a different type of the cells, but if you culture it for the 4-week time, right, then all the other cells will be died. So we have taken the advantage of the unique property of after 4-week time, we have 100% purity of the mast cells. No other cell will be able to survive. And then we look for the different type of the parts like the proliferation, the mechanism, and the other pathways. So here you can see the propium iodide assay, and in the propium that indicates the cell cycle study. Basically, in the cell cycle there are 2 important phases. One is called the subG1 phase that indicates the apoptosis, and G2 is the indication for proliferation, right? So here if you compare the vehicle treatment and IL-33 at different concentration, can you see any change in the subG1 population? This, there is no change, right? Means that IL-33 do not have any effect on the apoptosis. But if you see the G2 population here, can you see any change? Yes, there is a significant peak, right? So that means IL-33 is basically responsible for enhancement of the G2 phase, or in another word, we can say that IL-33 is responsible for the proliferation of the mast cells. So the same thing here you can see in the graphical form, that IL-33 is basically indicating the mast cell proliferation, and here this is a significant effect. Next question was the mechanism. So now we know that IL-33 is responsible for mast cell proliferation, but what is the signaling pathway? So for this study, we have used the ST2 knockout mice as well as the MYD88 knockout mice. And then we get the bone marrow derived mast cells from there, and then we have given the treatment of IL-33. And here you can see that IL-33 basically do not show any proliferation in ST2 knockout mice as well as in the MYD88 knockout mice. Similarly, when we look for the G2 population and subG1 population, again we could not see any change. So this result indicates that IL-33 needs this receptor ST2 and the MYD88, then only it will be able to show the proliferation. The next query was the signaling cascade. So you remember this picture that ST2 is responsible, MYD88 is responsible. Now, which MAP kinase is important in the cell proliferation? So for this study, we have treated the cells with the JNK inhibitor, ERK inhibitor, and P38 inhibitor, and then further challenge with IL-33. So here you can see first two graphs. This is the control and this is the IL-33 treatment. Very nice proliferation. But when you use the inhibitor here, you can see these three graphs are indicating the inhibitor part. Then you can see the inhibition in the proliferation in presence of JNK, ERK, and P38 MAPK. If you see the last graph like this, so you can see the significant change here. Like this is the MAPK, this is the MAPK, and this is the MAPK. So you can clearly see the significant inhibition when we have used the P38 MAPK inhibitor. So this result basically indicates that IL-33 could be the novel inducer of the mast cell proliferation. If you see the pathway, then basically it is governed through the ST2, MYD88, and P38 MAPK dependent pathway. And this finding may open the new perspective in case of understanding the IL-33 in the allergy and inflammation with respect to the mast cells. Not only in case of the asthma, but maybe there are several other diseases that are associated with the mast cells. So our objective is to target the mast cells or to target the interleukin-33 directly, because if you see the drugs nowadays for the asthma, do you know any drugs like antihistamine, right? So basically what happens that mast cell release the histamine, then we have the antihistamine. Mast cell release the leukotriene, so we have the anti-leukotriene. But there is not a single drug available in the market that can target the mast cells on the upstream pathway. So my hypothesis is if you target the IL-33 on the upstream, then cells will not be activated, they will not be released the mediator. So all the mediator release process will be stopped, and then there could not be any exacerbation in the asthma, because asthma is basically the airway hyperresponse. When then airway gets damaged, then IL-33 may comes out, then IL-33 can activate the mast cells, the mast cell will release their mediator, and that can further exacerbate the severity of asthma. So by targeting the interleukin-33, we can have the new drugs in case of the allergic diseases or the inflammatory diseases that are associated with the mast cells. So this is the one aspect of the study. Secondly, like what is the role of the IL-33 in the cancer? So as we discussed that IL-33 is a new molecule. It's a recently discovered molecules, and then there are a lot of researches going on in the several directions, like people are looking at the role of IL-33 in the autoimmune diseases, in the inflammatory diseases, in cancer, neurological diseases. So there are like a different type of disease. If you just search in the PubMed IL-33 and then you search your own area in which you are working, I'm sure you will get the many articles, and that could be, but it could be the novel also, because there is very limited research in the field of IL-33. So if you see the IL-33 in the cancer, then IL-33 could be the possible inducer and prognostic marker for the cancer development, with a direct effect on the tumor cell, promoting the tumorigenesis, proliferation, survival, and metastasis. IL-33 can also promote the tumor growth, metastasis by remodeling in the tumor microenvironment, that's called the TME, and they can also induce the angiogenesis. But the problem is there is no black and white area, right? Like you cannot say that IL-33 is the pro-tumorigenic or it is the anti-tumorigenic. Depend upon the condition, situation, the models, the human subjects. Like there are different type of the aspects, and there is a controversial studies are there, and there are very limited studies also at the same time. So IL-33 can act as a pro-tumorigenic also. so they can activate the different type of the cells like mast cells, T regulatory cells, neutrophils. They can release their cytokines, inflammatory molecules, and that they can go for the angiogenesis, invasion, tumor growth. But at the same time, they can also activate the NK cells. That play important role in the tumor regulation as a tumor rejection. So basically, NK cell and the cytotoxic cells, they can also be get activated by the IL-33. So now I think it is the need of the hour to investigate like the role of the IL-33, like how these cells, how these molecules are important in targeting the cancer, and which type of the cancer. As you know, like there are different type of the cancer. So maybe it could be the pro-tumorigenic in one type of the cancer and anti-tumorigenic in another type of the cancer. So there is a study is already published in the Frontiers in Immunology that they have looked for the interleukin-33 level in the normal tissue subjects and the breast cancer subjects. So they found the serum concentration of IL-33 was associated with the family history of the malignant tumor. Means the person who has a family history of the tumor, they have the IL-33 as compared to the person who don't have the history. So basically, increasing the tumor or enhancing the cancer. When they look for the immunohistochemistry of the interleukin-33 in the breast cancer tissues and the normal tissues, here you can see the high expression of the IL-33 in the breast cancer tissues and the normal tissues. So there is very limited reports available in the area of the IL-33 in the cancer. But yes, we can say that the IL-33 may play important role in the progress of the breast cancer and may be the useful biomarker for predicting the progress and the metastasis of the breast cancer. Not only for the breast cancer, it should also be investigated in different other type of the cancer. So thank you very much for your kind attention and patient listening. So I'm also planning to extend my study in the field of cancer also, and since it's a good opportunity for me to interact with you guys who are the cancer experts. So if someone is interested for having that collaboration with me, I would be more than happy to have it. I have already given my phone number as well as the mobile number. So if anyone is interested, please feel free to talk to me at any time. Yeah, thank you very much for your kind attention.
Thank you very much,
Yes,
sir.
so if there is any question, then please feel free to ask.
Thank you very much, sir, for your valuable information regarding cancer and its biomarkers. It will definitely help us further. So now the session is open for the question answer round.
Hello, Rohit, Dr. Rohit.
Yes.
Very nice presentation. But my query is just to understand why you have taken bone marrow derived mast cell. Why can't you choose because mast cells generally in the mucosal immune system. So why can't you choose the cell lines related to the respiratory pathway or the mucosal cell lines rather than the breast bone cells? Is there any specificity, or can you explain some light on this? Yeah.
So basically this bone marrow derived cells, bone marrow, it's basically experiment was on the mice. So we have taken the bones from the mice. It's not from the human subject.
Okay.
As we have discussed that you cannot get the mast cell from the blood. So it is difficult to get the mast cells when there is any operation or any skin removal, then definitely we have the opportunity to get the mast cell from there. Or if there is any pregnancy case or in the delivery, if there is a cord blood, so you can also get the mast cell from the cord blood.
Okay.
So these are the two important sources for the mast cells if you are working in the human subject. But definitely mice, I think it's a very nice model. So we can get the bone marrow derived mast cells. As far as the cell lines are concerned, then we have the HMC cells. That's called the HMC, like the human mast cells. So that is basically available in the form of the cell lines also. So we already have this cells in our laboratory.
Okay.
But we have not investigated the role of IL-33 on HMC1. So there is another project going on in this area.
Yeah, nice. Thank you. Is there is no phenotypic difference between these two cell lines, na? Means whether it is blood or bone marrow derived mast cells? No phenotypic difference.
There is only one phenotype in the bone marrow derived mast cells. Other than the bone marrow derived mast cells, you can also get the connective tissue like mast cells and the mucosal type of the mast cells. It is respective to the MCT, that is the tryptase containing mast cells, and MCTC, that is the tryptase and chymase containing mast cells. So if you see heterogeneity in the human, we have the two different types of the mast cell. One is called the tryptase MCT, T means tryptase and MCTC is tryptase and chymase. So basically connective tissue mast cells is basically the MCTC type, and MCT is basically the mucosal mast cells.
Right. Thank you, Rohit.
I would request those who have to give their oral presentations, please go to B1007 room number.
Yes, if there is any other questions.
And for posters, please go to B1 block.
So I think there is no more questions, but just before leaving the meeting, I would suggest the student, because my basic presentation was just to introduce the I-33 with you. So if you are working in any area, you just look for your area and I-33. I'm sure you will get some interesting topic there. So if you are looking for your MSc topic or PhD topic, I think that could be really helpful for you.
Okay, Dr. Saluja, on behalf of all the student and faculty, I thank you for the wonderful talk. And it's nice to see you, Rohit. I'm Sarika.
Okay.
So Rohit
it's indicating
is my...
that Dr. Sapna Sharma.
Yes, because that is from login from her ID. So,
yeah, it's nice to hear from you after a long time.
Yeah, it's very long time. So I would like to tell all the person here, he's my lab mate. So we did the PhD work together. So it's very nice to see you. Okay, thanks a lot, Dr. Rohit, for a very nice talk.
Thank you, madam. Thank you.
Thank you. So now, yeah.
Okay, so now I am leaving the meeting. Thank you.
Okay, thank you, Rohit. Thank you.
Now I would like to introduce our next speaker for the session, Dr. Ejaz Ahmed Bhatt. He is currently working as a research scientist at Department of Translational Medicine, Sidra Medical and Research Center, Doha, Qatar. He completed his PhD from one of the premier institutes, All India Institute of Medical Sciences. After his PhD, he moved to Vanderbilt University Medical School, Nashville, USA, for postdoctoral fellowship. During his 8 years of postdoctoral training at Vanderbilt, he has gained wide experience in the field of cancer biology. His focus of research was on the role and regulation of tight junction proteins, claudins, in colorectal cancer and the role of bile acid-induced APE1/REF1 in esophageal adenocarcinoma. He has published in well-reputed journals like Nature Communications, Oncogene, Cancer Research, Molecular Cancer, and many more. Now I would request Dr. Bhatt to proceed with his talk.
Yeah, hello.
Yes, sir.
Am I audible?
Yes, sir, you are.
Okay, yes.
Hello, Dr. Bhatt.
Yeah, good afternoon, everyone. I think it's a good afternoon there, but for me it is still a good morning. So first of all, I would like to thank. I think there is some background noise coming from somewhere. I don't know where is it.
I think Dr. Bhatt, please open the camera.
Okay, is off. Okay.
Yes. Thank you. Yeah, now we can see you.
Okay, so now it's fine.
So I would like to request you please be on time because we are already very late in
yeah, that because even I'm late for the other presentation. So how much time is for me now? Because I will do accordingly.
It's 20 minutes, I think.
30 minutes?
20 minutes.
20 minutes?
20 and 5 minutes for question and answer. 25 minutes.
Oh, isn't that too less? I will try, because
we apologize.
The presentation was for like I think 25 minutes, but we'll try to finish it fast. So I think better I start right away rather than wasting more time here. So I will share my screen here. You just can see it, I think. So just let me know if it is. Can you
Yes, sir. It's
So is it visible?
yes, sir. It is.
I just need to, I want to delete, have the screen where I can, I don't know, why is it coming the other side? Just, how can I need to, I want to have the full screen for this. You have any idea how can I go to the hide screen from there? I don't know. Hello?
Yes, sir.
I want to take, I want to have a full screen because I don't know how can I do it, because the problem is that it's showing me that other slide which I don't want it there, so that
Sir, if you could restart once, maybe.
no, it was, I think,
So I think there's an option of slideshow icon. It's showing beneath, like. Srati down, it's an icon.
maybe. I don't know.
Sir, you can continue.
I think that should be fine, yes?
Yes,
Is
sir.
it fine?
Yes, sir, it's fine. It's visible.
It's visible, yes? Okay, okay. So I will start then. First of all, I would like to thank organizing committee and especially Dr. Tabish for inviting me as a speaker for this conference. My title talk for today is epithelial barriers, tight junctions and disease. What role do claudins play? Before going to the whole details of this topic, I would like to give a brief introduction so that it becomes easy to understand the whole structure. As I have mentioned here, that strong fences keep the enemies away. So these epithelium are these epithelial barriers. So these epithelial barriers are important for so many, they perform so many functions, especially if we are talking about intestinal epithelia, airway epithelia, or gastrointestinal tract, or the skin epithelia. So they have multiple functions, and especially the important function is the nutrient uptake, transport of ions, molecules, and also mechanical integrity to cells, and also important is providing rigidity to the epithelia. So these epithelial cells which form the epithelium are held together by tight junctions, which led them to be together and then help them in performing so many other functions. So here talking about the tight junctions, there are 4 main tight junctions, which are adherent junctions, caterins, and then desmosomes and gap junctions. In my talk, I will be talking about, because in the tight junctions, there are 3 important family of proteins, which are junction adhesion molecules. Then we have claudin proteins, and then we have the other class, which is occludins. So these are 3 family of proteins which are important for the tight junction structure and function. So among the tight junction proteins, I will be focusing on claudins. So if you see a structure here, so these are the epithelial cells together held by these are different like tight junctions which have the proteins called as claudin proteins. And then you see the other junctional adhesion molecules, caterins. These all are form the tight junctions, and which help and then if you see the side here, the epithelial cell. So here you can see that the tight junctions keep the cells together, And the important function they also do is they give polarity to the cell, that they give apical and basal surfaces to the cell, and thus help in giving polarity to the cells. And besides that, they help in absorption, transport of nutrients, and they are important for the barrier functions. So the stronger the tight junctions, better will be the protection from microbial antigens or any other of the contaminants. So if we go to the next slide, here you can see that epithelia is everywhere in the mammalian body. You can see so many epithelia, which so all have an important function, which I said it could be either the protection, or it could be the absorption, it could be the transport, or it could be the signaling communication. And then here you see that, as I mentioned, that 4 important type of junction proteins, which are occlutins, tricellulin, claudins, and junctional adhesion molecules. So these are important proteins which form the tight junctions, and tight junctions in turn help in performing so many other functions, especially they are main regulators for the epithelial barrier function. They are responsible for the apical cell-to-cell contacts. They are important for maintaining cell polarity, and they are also important for the paracellular permeability, which is the barrier function for the tight junction. So you can see the structure. This is just the structure. And then the tight junctions are very complex, and they are very dynamic structures. As you can see from the whole range of proteins here, or the signaling molecules here, which are involved in so many essential functions performed by these tight junctions in the body. And then you can see, if we talk about a single epithelial cell, the structure of the epithelial cell here, you can see these apical and the basal surfaces, and then you see the tight junctions. These tight junctions are responsible for giving the polarity to the cell, and then in addition to performing the other functions, as I mentioned, the important is the fence function, that they prevent the intermixing of components between the apical and the basolateral membranes, and also help in maintain the polarity. So this is how it works, structure. So then comes the other important function, which is, as I mentioned, that if you see the structure here, you see that when the intestinal epithelial barrier is very intact, so it prevents from it crossing all other drugs, antibiotics, or allergens to cross, and then keep the help in intestinal homeostasis, which is also important function for these tight junctions. And if there is a compromised intestinal barrier, you can see that these tight junctions get disrupted, and then they result in the transport of so much bad things inside the interstitial spaces, resulting in inflammation and other diseases. And also one of them is also the cancer. So now, since, as I mentioned, that tight junction 4, but I will be talking more of claudins here. So claudins are a group of 20 family members, around 20 proteins, which have 20 to 27 kD molecular weight. They are tetraspanin proteins, which have N and C terminal domains inside the cytoplasm. And then they are differentially expressed in different tissues, and then perform the specific functions for each of the tissue. And so these claudins are being regulated by, so that was the structure, then the function, and then the regulation. So these are regulated by transcription factors like CDX1, 2, GATA4, HNF, then cytokines, growth factor signals, and also other like the metabolic acidosis and the vitamin D receptors and all that. They regulate the claudin expression. So if you can see that here, this was the paper from our lab where we showed that this claudin-1 expression in colon cancer cells is being regulated by the transcription factor, which is a caudal homobax protein CDX2, which cooperates with Wnt pathway to regulate its expression. And then we also showed that this claudin-1, which is an important tight junction protein, is helps in epithelial to mesenchymal transition, which is important phenomena for cell transformation, or you can say cancer or the other process like metastasis or invasion. The other important function for clot, as we saw that in one of the reviews published, where we showed that these tight junction proteins are also important for the signaling pathways in inflammation as well as in cancer. So the expression levels have been implicated in a number of cancers, which I said is a tissue-specific manner. Suppose in case Claudin-7, a decreased expression has been correlated with invasive breast cancer, while as Claudin-4 in pancreatic cells, Claudin-3 and 4 upregulated in ovarian and breast cancer. So here is a list which shows that these proteins, these Claudins, which are around 24 members, they change in a specific manner in different cancers. Like in some they are upregulated and in some they are downregulated. So I will be more focusing here on Claudin-1 and Claudin-7, though we work on almost all the Claudins, but we'll be talking here more on Claudin-1 and Claudin-7. So as Claudin-1 is important as for the normal function, is very important because studies have shown that Claudin-1 knockout mice dies after 1 day of birth, is downregulated in breast cancer, upregulated in pancreatic cancer. And then, so this was the paper published in JCI, where we have showed that this Claudin-1 expression is increased in colon cancer and also in the metastasis. So if you see that these are the colorectal cancer cells, and these are highly metastatic 486-26, you can see Claudin-1 significantly upregulated, and then Claudin-7 is down there. And these are the primary and metastatic patient samples where you can see increased Claudin-1 expression and a decrease in Claudin-7 expression. So here we show that this Claudin-1 is increased in colon cancer and is responsible for metastasis and also for cell migration, invasion, and other important functions. So it is upregulated in colorectal cancer. And then the other important role for Claudin-1, if we talk about that, is the localization of Claudin-1, whether it is a cytoplasmic, whether it is a nuclear, or whether it is a membrane localization, because that is also very important for the functioning of all of the tight junction proteins, especially the Claudins, where we see that it is mislocalized in cancer. You can see in nuclear, and then here you can see Claudin-4, which is membranes. And then you see lymph node and liver metastasis, higher expression of Claudin-1. In order to prove that concept that Claudin-1 is involved in intestinal homeostasis and all that, so we constructed a Claudin-1 transgenic mouse model, and these are where we have shown that you can see that the transgenic 1 expresses higher expression of Claudin-1 in intestine colon, and in case of colon for a transgenic mouse, you see a decrease in the goblet cell number. And these are showing that Claudin-1 is, expressed there. It is there in the transgenic mouse model. And again, here you see that Claudin-7, a decrease in Claudin-7, which shows a reciprocal, correlation between Claudin-1 and Claudin-7, which I will come later. And these are the colon tissues from the mice showing that, yes, we have the Claudin-1 transgenic mouse. And then, as we said, that this Claudin-1 regulates intestinal homeostasis through the notch signaling. So if you see the transgenic mouse model, we have Claudin-1, and then you see that notch intracellular domain increased expression. And these are the markers, HES1 for this notch signaling, and then MAT1 is the opposite for that. So you see an increase in HES1 and decrease in MAT1 expression. And then these are the Claudin-1 expressing colorectal cancer cell line as W480, where you have higher Claudin-1, and then you have notch domain increase, and along with the phospho-ERK signaling. So that was about the Claudin-1 for regulating intestinal homeostasis through the modulation of notch signaling. Then we had published one paper that was about Claudin-1 increases colon tumorigenesis and decreases survival, where we again used a claudin-1 transgenic mouse model, which was crossed with APC min mouse to have a model for, because APC mutation is responsible for also cancer and also other adenoma formation here. So if you can see that this is the APC1, and when we have APC1 and claudin-1 together, we can see that increase in the number of tumors in the intestine of claudin-1 transgenic mouse. Then you can see from the histology that this is glandular, more number of this hyperplasia and all that. Then you see invasive carcinomas in case of claudin-1 transgenic mouse models. And when we did microarray analysis of these, we found that claudin-1 overexpression is involved in notch signaling, Wnt signaling, inflammation, and barrier functions. Interestingly, we found that claudin-7 was downregulated in these claudin-1 expressing tumors are in the claudin-1 transgenic mouse models. So then, since that was about the claudin-1, since the interesting thing that we found was the claudin-7 going down with the upregulation of claudin-1 or in the claudin-1 tumors, there was less expression. So we thought that, so concluding that claudin-1 is a tumor promoter in colorectal cancer, and then thinking that maybe claudin-7 has an opposite role of claudin-1. So then we targeted claudin-7, thinking that it could be a tumor suppressor. And then we showed that when we have higher expression of claudin-7, these are the clones that overexpress claudin-7, 620 cell line, which doesn't express the claudin-7, we overexpress it there. And then we found that claudin-1 going down and ecadherin, which is an epithelial marker, going up, and all mesenchymal markers, they go down. And if we knock down claudin-7 in two cell lines, HG29, DLD1, we see that claudin-7 knocked down, but claudin-1 coming up, ecadherin going down, and other mesenchymal markers coming up, which shows that if you see the staining here also, that you see that increase in ecadherin expression, claudin-7, and also decrease in other ZO1, which is a mesenchymal marker. So showing that claudin-7 is responsible for reversing of mesenchymal to epithelial transition, or you can say that it inhibits epithelial to mesenchymal transition, because EMT is a very strong phenomena in cancer transformation or in cancer metastasis. So with that, we showed that claudin-7 is a tumor suppressor. And then we did, using some TCGA data, where we again showed that claudin-7 is downregulated in the colorectal adenocarcinoma samples compared to the normal. And then going for the survival, which shows that claudin-7 expression, again, the colorectal adenocarcinoma patients, where we show that low claudin-7 expression is associated with poor survival. And then using these different stages of the colorectal cancer tumors, and we found that claudin-7, if you see this normal, is high, but as we progress from adenoma to other stages of the colorectal cancer, so this claudin-7 is reduced in there. So that was showing that claudin-7 is involved in epithelial to mesenchymal transition and is a tumor suppressor. Then we showed that was the paper published in Oncogene. Then continuing with that study, we thought that maybe then we could change or modulate claudin-7 expression through different means. And then we found that when colorectal cancer cells were treated with TNF-alpha, which is a pro-inflammatory cytokine, and DSS is a dextran sodium sulfate, which is used as for inducing colitis for inflammation model, that it irritates intestine and leads to inflammation, which is also considered as one of the most widely used mouse model for colitis. So if you can see that claudin-7 expression with the TNF-alpha DSS, it goes down. And here you can see that claudin-7 expression going down with TNF and with the treatment of DSS, which are both responsible for inflammation. And then we see that claudin-7 expression increases the anti-proliferative effects of cisplatin, as we know that cisplatin 5-FU, these are the drugs that are used for chemotherapeutic drugs for the colorectal cancer. So we thought that maybe Claudin-7 expression will increase the effectiveness of these drugs, or maybe acting in a synergistic manner there. And then we found that when we have Claudin-7 overexpression, so you can see that the cell viability is with the cisplatin or with the 5-FU significantly going down. And when we have a knockdown of Claudin-7, so you can see that with the cisplatin or with the 5-FU, it goes up. So which means that Claudin-7 expression, it plays a role in increasing the effectiveness of cisplatin and 5-FU in inhibiting the cell proliferation. So then we looked for its effects on apoptosis, whether we could have more apoptosis while using Claudin-7 expression with cisplatin and with 5-FU. And if you can see that, these are clone 1 and 2 which overexpress Claudin-7. And then you can see that with the increase in cleaved Caspase-3, which is an apoptotic marker, so with the cisplatin with combination, so you can see a significant increase in cleaved Caspase-3, and also with the treatment of 5-fluorouracil, which shows that yes, this Claudin-7 plays an important role in increasing the effectiveness of cisplatin and 5-F in inducing apoptosis and inhibiting inflammation. Next, we wanted to, as I mentioned, that the barrier function is the important function for tight junctions, which means that these tight junctions, if they are deregulated or there is any disruption, so that leaks a leaky epithelium, or you can say a leaky gut, which results in the free transport of ions, molecules, water, or any of the antigens or any of the microbial contaminants. So it is very important that these tight junctions need to be very tight, they need to be very adherent to each other, and they need to be very tightly regulated so that they can regulate the transport of molecules or ions across the intracellular epithelia. So here you can see transepithelial electric resistance and these are the two parameters that are important for measuring the integrity of epithelial integrity of the cells. And here we see that when we have overexpression of Claudin-7 in 620 cells, you see that at 48 out of 70, there is an increase in transepithelial electrical resistance. So which means that these are tightly held together, these epithelial cells, and then they don't result in the free transport of molecules or ions, which is important for maintaining the integrity of the cells. So you can see that increased transepithelial electrical resistance. And then you see that when you have increase and when you have a knockdown, you see a decrease in this transepithelial electrical resistance. And this should go opposite, the paracellular transport, or you can say the paracellular permeability, because when the junctions are tight, so there will be no free transport, which means the permeability will be less when you have an overexpression of Claudin-7, which shows here in 620. But when you have a knockdown of Claudin-7, so it increases because the tight junctions are not firm, or they are disrupted, and that result in the free transport there. And then we, because these are different, we were trying different means to have increased expression of Claudin-7 and decreased expression of Claudin-1, as mentioned, Claudin-1 as a tumor promoter and Claudin-7 as a tumor suppressor. So then we used these are the recent work that is going on, where we show that the curcumin, which we consider as a protective, so many papers, so many literature for curcumin as a protection for so many diseases, including cancer, and then chemo drugs, which is cisplatin and 5, which are already there. So when we treated cells, colorectal cancer cells with curcumin and these, we saw that you can see that there is an increase in Claudin-7 expression with curcumin, with cisplatin, with 5-FU at 10 millimolar and all that. So which means that one of the mechanisms could be the increase in expression of Claudin-7 for this. So this is the work that is in progress, which show how Claudin-7, because knowing the mechanism, how it increases chemosensitivity to cisplatin, 5-FU, then if Claudin-7 has any role in metabolic regulation, because there are few studies which are digging into the metabolic role of Claudin-7, we have some preliminary data for that too. And then if you has any role for cancer stem cell regulation. We have some preliminary data for this that yes, it decreases the expression of cancer stem cell markers when we have increased expression of Claudin-7. And then if Claudin-7 is epigenetically regulated in colorectal cancer, because in colorectal cancer, we have shown that in colorectal cancer cells, this is in some of them which are highly metastatic cells, regulated. So which means there is some mechanism which is regulating its expression, maybe epigenetic modification or any of the methylation that is responsible for its down regulation. So that is also in process using certain demethylating drugs and then looking for the expression of Claudin-7 and then this Claudin-7 expression in human colorectal cancer. So the long-term goal, because based on these results, is interesting, very straightforward. We say that maybe it could serve as any type of biomarker in the future, either a prognostic, diagnostic, or it could be a response marker. So at the end, we'd like to acknowledge many people, not especially Sidra Medicine team members, which is Dr. Haris, Tariq Masood, which is staff scientist in our, then Sabah Nisar Shima, these are master students, and Sumaya, she's an extern. Then the core people, program. We have a collaboration with here, especially with Dr. Shahabuddin, and external collaborations I have with my previous labs, which are still going on. Punita Dhawan, Vanderbilt University, then my second PI, postdoctoral PI, Dr. Wael Rifai, which is now in University of Miami. And then Dr. Puneet, which is at St. Jude Children's Hospital, Dr. Ravinder Reddy, which is at Penn State, Dr. Muzaffar from IUST Kashmir, Islamic University of Science and Technology, then Dr. Mayank Singh, which is from AIIMS Delhi. So would like to conclude here, and thank you very much for now.
Hello. Thank you, Dr. Bhatt, for a very wonderful talk. And now the session is open for question. If the audience have any question, then please ask. So if there is no question, I would like to, sorry.
Very nice presentation, Bhatt sir.
Thank you very much.
So I have one or two for my clarification only. So this
Yes, sure.
Claudin-7 molecule is related to any metastasis type? Do they have any role in metastasis? Because as you told, the Claudin-7 expression was decreased when the color CRC is increased. So did you have, related to the metastasis?
Yes, actually, because when we have these metastatic, we didn't have the direct proof here, but based on the data that we have from Claudin-1 transgenic mouse models, where we had, all liver metastasis samples. So there we saw that, Claudin-1 Then we did some migration invasion assays with the overexpression of Claudin-7. So we found that it is involved in inhibiting cell migration, cell invasion, and also the clonogenic transformation. So all which are involved in the metastasis. And then when we did the xenograft model for Claudin-7, so we found that because since when we used the because we had the overexpression of Claudin-7 there, so we found that the tumors were completely gone when we had Claudin-7, which means that because we didn't have any way to look for the metastasis because the tumors were not growing in presence of Claudin-7. So which means that it could have a role in inhibiting metastasis as well.
One more thing hai, suppose if we think about the microRNAs, so in your study if you add any microRNA expressions, overexpression may be related to the claudin 7 downregulation. I don't know, means just I'm
yeah, it's a very good question. Yeah, so because since time we didn't, so actually we have some data where we are, yes, we have about some of the microRNAs that regulate the expression of few of the collagens. So we are digging deep into that. That could be also one of the mechanisms where we say that okay, the expression of these is regulated by microorganisms, these microRNAs, and in addition to other epigenetic modifications.
Okay, thank you. Over to Chairman.
Thank you very much.
Thank you, Dr. Bhatt. So I think now we can go ahead for another talk. Thank you so much.
Thank you very much. Thank you. Thanks, Dr. Tabish, and thanks organizers for this. Thank you very much.
Thanks.
Bye. Good luck.
Now I would like to invite our next speaker for the session, Dr. Sujata Pathak. She is a scientist in preventive oncology at All India Institute of Medical Sciences, Delhi. She has completed her PhD from AIIMS, Delhi as well. Her research studies areas include molecular biology, biochemistry, tissue culture, immunohistochemistry. Now I would welcome ma'am to proceed with the talk.
Thank you, Dr. Sapna. And first of all, I would like to thank Dr. Tabish and the organizers for inviting me to deliver the talk. So I'll start sharing my PPT. Is it visible?
Yes, ma'am, it is.
All right. So today I'll be going to talk about cervical cancer. And today I'm going to present a part of my work, which is association of role of 1-carbon metabolism in progression of cervical cancer. So first, let's understand the magnitude of the problem or the burden of cervical cancer worldwide and especially in our country, India. So worldwide, according to Globocon 2020, cervical cancer is the 4th most common cancer and the 4th most leading cause of death in women worldwide. With 604,000 new cases diagnosed every year and 342,000 deaths per year. Lower middle income countries are comprises higher burden of this disease, and in India alone, which contributes to around 21% of the total cervical cancer burden of the world. It has 123,907 cases reported every year with 77,348 deaths. And so it is very sad to know that one woman dies of cervical cancer every 7 minutes in our country. So this is the pie chart showing distribution of cases of deaths for the top 10 most common cancers in 2020 by Globocon. So you can see here that mortality and incidence wise, it is the 4th most common cancer worldwide. So what causes cervical cancer? So there is a virus called human papilloma virus, which is a positive agent for causing cervical cancer. So it is a double-stranded DNA virus, approximately of 8 kb size, and is usually classified into high risk and low risk HPV types depending on their oncogenic potential. And there are over 200 subtypes of this HPV is known. And out of all these HPV, HPV 16 subtype is the most oncogenic type. And along with HPV 18, it alone accounts for approximately 70% of the cervical cancer worldwide. And in India, it has been shown that around 82% cancer, cervical cancer, women have this HPV 16 or 18 infection. So this is the diagram showing how HPV infection occurs in women and how it is getting cleared. So first, the HPV virus infects the normal cervix, and most of the time, 90% of the women clear this HPV infection by their own immunity. So it can be reversed here. You can be HPV infected and you are free from HPV infection without even knowing. So, but rest 10% may progress to precancerous lesion where it is still not a cancer. So from there, if the HPV infection persists, because it is not only infection that causes cervical cancer, the infection has to be persistent to cause cervical cancer. So less than 1% of these HPV infection finally causes invasive cervical cancer. So as I have told you in the previous slide that cervical cancer pathogenesis is caused by oncogenic high-risk human papilloma virus infection. And not all women infected with HPV develop cervical cancer, indicating other factors may involve along with HPV in inducing cervical carcinogenesis, meaning making this HPV infection more persistent towards developing cervical cancer. And the good news is that this cancer is a slow growing cancer. It generally takes around 10 to 15 years to get cervical cancer from the day you have got infected. And out of those also 90% is already generally cleared by your immunity. But still there is a long, very wide window for this to progress towards cancer. So this large window, this wide window through from HPV infection to precancerous lesion to cervical cancer provides us opportunity for prevention, early detection, and better treatment. So what are the risk factors which increases the persistence of this virus in the cervix? So here are few major risk factors, including multiple sexual partners, as it is a sexually transmitted disease, smoking, multiparity, long-term use of oral contraceptive pills, HIV infection, and lower or poor socioeconomic status, and poor nutrition or diet. So I'll be focusing on this portion of the risk factor, dietary part. So this is a recent paper. We know that this is a very famous Hanahan, which they are publishing this paper on hallmarks of cancer since last 20 years. So this is the recent update of hallmarks of cancer, where they have added this epigenetic reprogramming also in hallmark of cancer. And in this presentation, I'll be also focusing on deregulation of cellular metabolism as well as epigenetic reprogramming. So don't worry about this picture. This is just a snapshot of a part of a metabolism which is occurring in our body. So my intent of showing this picture is just to let you know that all these complex metabolism are there for few functions, which is providing energy, immune functions, cognitive functions, and these all functions are carried out by an important carbon donor, which plays a very important role in DNA synthesis, repair, and methylation. So, 1-carbon metabolism comprises a series of interlinking metabolic pathways that includes the methionine and folate cycle. This methionine, essential amino acid, and folate cycles that are central to cellular functions. So providing 1-carbon units, that is methyl units, for DNA synthesis, amino acid synthesis, polyamine, and phospholipids. So generally, primarily, 1-carbon methyl units enter the cycle as this 5,10 tetrahydrofolate. So if there is a deficiency of folate, there will be a disruption of this cycle, and these processes of DNA synthesis, repair, and methylation will be affected. So we just wanted to check whether a less efficient 1-carbon metabolism increases cervical cancer risk or not. So the objectives were to study the level of cobalamin. I'm sorry, I didn't talk about cobalamin. Here you can see that in this cycle, we also need methylcobalamin, which is vitamin B12, for conversion of homocysteine to methionine. So the objectives were to study the levels of cobalamin, folate, and homocysteine in serum of normal, stellar precancerous lesion, and cervical cancer patients. And second objective was to study the presence of HPV and subtype 16 and 18 in cervical biopsy samples of these subjects and correlate them with the serum folate status. And the last objective was to study the methylation status of genes involved in cervical carcinogenesis, such as CTH1, retinoic acid receptor beta, and HIC1. So this is the overview of methodology. We collected total of 105 cervical samples, out of which 35 were normal, 32 were precancerous or SILS, and 38 patients were of cervical cancer. So from all these subjects, tissue as well as blood was collected, and from tissue, we have done HPV testing and type-specific HPV testing, and also we have done methylation-specific PCR for any epigenetic or hypermethylation of promoter region in the genes, which I have shown you in my objective slide. And from blood, we have measured folic acid, vitamin B12, and homocysteine. So these are the clinical pathological features of patients. So this table is showing that mean age for normal were 45 plus 6.5 years, precancerous 39.9.8 years, and out of cervical cancer, how many were well-differentiated squamous cell carcinoma, moderately differentiated squamous cell carcinoma, and poorly differentiated squamous cell carcinoma. And these are the clinical stages where we have collected the samples from these women. So this is the result showing HPV status in normal, precancerous or SILS and cervical cancer cases. So we found that out of all these 105 samples, in normal, we found 8 HPV positive women, and 25 out of 32, which makes it 78% of precancerous women were HPV positive, and 92% we found in cervical cancer cases, women were HPV positive. And this is the just how these HPV women were divided in 16 and 18 positivity. And the next table is showing the level of folate, vitamin B12, and homocysteine levels in these three categories of women. So we found a significant difference between folate, vitamin B12, and homocysteine levels amongst these three groups. In case of folate and vitamin B12, we found that it was significantly reduced. These levels were reduced in cervical cancer patients in comparison to cells and normals. And homocysteine levels were increased, significantly increased in comparison to normals. So this is the just a plot showing correlation between vitamin B12, homocysteine. We can see that this is an inverse correlation between vitamin B12 and folate with homocysteine. So from this part of study, we concluded that women which are diagnosed with SIL and cervical cancer had a significant lower serum folate and vitamin B12 status than those with normal cervical cytology. Whereas level of homocysteine was significantly higher in serum of women diagnosed with SIL and cervical cancer than those with normal cytology, histology. Also, serum folate and vitamin B12 levels were significantly lower in women with HPV infection than HPV negative samples, whereas the vice versa was seen for homocysteine levels. So, DNA methylation is an epigenetic mechanism that regulates chromosomal stability and gene expression. Various studies suggested that hypermethylation of certain tumor suppressor genes may be related to the development of cervical cancer. So we wanted to see whether aberrant DNA methylation is involved in this disruption of 1-carbon metabolism. So this is a slide showing about diet and epigenetics. Nutrients and bioactive food components can modify epigenetic phenomena and alter the expression of genes at the transcriptional level. Nutrients can reverse or change epigenetic phenomena, thereby modifying the expression of critical genes which are associated with physiological and pathological process, including embryonic development, aging, and carcinogenesis. So this is just a chart to show that if we follow the wrong diet, poor diet, or the safe Mediterranean diet, how our epigenetic can be involved and results in the kind of obese or lean phenotype according to our diet. And this obese phenotype can lead to disease phenotype, and it can be inherited in the next generation as well, and hence the increasing the risk of disease. So we just wanted to check whether folate status and aberrant DNA methylation show a progressive change across stages of cervical pathology from normal cell to SIL to cervical cancer. So for this we have done methylation specific PCR. This is just a flow chart of the methodology. And we have done CDH1 gene, ECADRine, which acts as a transmembrane glycoprotein that is important in epithelial cell-cell interaction. And also it acts as a tumor suppressor genes that prevent cells from growing and dividing in an uncontrolled way to form cancerous tumors. So this was the result of methylation specific PCR for CDH1. These are the findings which shows that we found only 1 out of 35 normal samples was methylated, and which makes it only 2.385%, and 21.87% of SIL samples were methylated, whereas there was a significant increase in methylation, promoter methylation in case of cervical cancer samples for this CDH1 gene. Next was retinoic acid receptor beta gene. This is also a tumor suppressor gene which is involved in embryonic growth, embryonic morphogenesis, cell growth, and differentiation. So what we found for this gene that 11% of normal tissue samples were methylated for this gene, but there was a significant difference between SIL and cervical cancer samples, which were methylated for this gene 56 and around 58% respectively. The last tumor suppressor genes we studied was HIC1 gene, which is an essential gene for mammalian development and has been found to epigenetically inactivated in many human cancers. So here we found that after doing methylation specific PCR, that in comparison to normal, it showed a significant increase in promoter hypermethylation of HIC1 in SIL and cervical cancer samples. So this is the consolidated table for all these three genes for promoter hypermethylation across all the three groups. So we found that in cervical cancer, for any gene, I mean for any of the three genes, we found that there was a significant increase in SIL as well as cervical cancer cases. Hypermethylation of any of these genes is fairly increased, which may, because of this frequency of promoter hypermethylation for all these three tumor suppressor genes, was increased, hence showing an increasing trend from normal to SIL to invasive cervical cancer specimen. And it can be used as a biomarker because it is already increasing so much in precancerous disease also. So the panel of these three genes may be used as a biomarker for identification of early cancer so that it can be treated before it becomes cancerous. So the conclusion from this presentation, this study was that role of folate, it shows a role of folate in modulating the risk of cervical cancer. Poor folate status contribute to cervical cancer through effect on 1-carbon metabolism via folate and homocysteine and DNA methylation. So maybe supplementation with folate and vitamin B12 could be a viable non-vaccine approach for cervical cancer prevention. And these three tumor suppressor genes, CDH1, HIC1, and retinoic acid receptor beta genes can be used as potential biomarkers for cervical cancer risk assessment. So this is the kind of model I prepared from my studies, that initially I showed you that HPV infection HPV infects normal cervix, and then 90% of the cases are transient infection which are being cleared by our immunity, and rest can progress to lower grade of SIL or higher grade of SIL, and then it can, and see the timeline, you can see that from infection, clearing the infection generally takes 18 to 24 months, or from this infection to become a fully fledged cervical cancer, it takes around like 12 to 15 years, which is too long, which is a very wide window for intervention. So, and in our case, nutritional factors, which are vitamin B12, folate or vitamin B9 or homocysteine, can here we can intervene here and can change revert this precancerous lesion to normal cervix, or DNA methylation also works at this level. From it is still there is a chance of regression. So if these things taken into account, here intervention can be done, and early detection biomarkers in the form of these promoter methylation tumor suppressor genes can be used. So these are the overall nutritional factors which are influencing HPV persistence, which is very, very or key importance for cervical cancer progression. Thank you. So this is just a message to show that people should undergo HPV test, pap smear test, or get vaccinated themselves if they are in 9 to 16 or 18 years of age. So this is just to promote this thing. Thank you. Any question?
Thank you, Dr. Pathak, for a very informative talk. So now the session is open for question. If there is any question.
Very nice talk, Sujata. But one thing I would like to ask, you told about the hypermethylations, right?
Yeah.
So
yes.
So did you get the sequenced the primers you have done to know the status of methylations? Because CPG islands conversions bisulfite after bisulfite conversions.
We didn't go for sequencing, but it was just a semi-quantitative thing. So to see if there is association, or we were just saying it is a yes or no phenomenon, that if there is a presence of hypermethylation or not. We have not done.
Actually, because we also worked for the CRC in the similar lines, where after sequencing we were able to know that more hypermethylations were observed. Okay, thank you, thank you.
Thank you, madam. I would also like to ask one question. You have suggested that there are 3 biomarkers you are suggesting that can be used to diagnose the cervical cancer. So I would like to know whether those can be used in clinical settings or still they need some more exploration.
Yes, of course, in going to human settings, it requires a lot of trials and all. This was just on research basis. So it was just suggestive of anyone can pursue it further, but we have done it on research basis that this is the study we have planned, and we have suggested that it can be done. So it cannot be done like from research. You cannot go directly to the clinic. It needs a lot of trial phases, different phases of trials to go through this.
And madam, what was the sample size?
105 total samples, including normal cells, I mean precancerous cells, and cervical cancer.
Okay.
105.
Thank you, madam. On behalf of all the members, I am thanking you for a nice lecture. Thank you.
Thank you.
Thank you.
With this, we conclude the current session. Now I would like to invite all the guests for lunch. Thank you.
पता लगा बाबू भी वही लटक गए। कहाँ से आए? मैडम ने भी देख लिया एक साथ चक्कर में बंद कर रखे बढ़िया गार्डन बढ़िया बना रही है। क्योंकि कंपटीशन नहीं है। हमें लगता है वो वाला लिफ्टिंग प्लास्टिक प्लास्टिक और एक कुछ पान ड्रिंक भी अब कल ये लग गया बाहर लगेगा नहीं लग रहा है। मैं मोदी साहब को बुलाता हूँ। हाँ आपको मिला नहीं मिला कभी है ना ये भी है सार को ये भी तो सोचो ये भी सोचो कि साथ भी इतने दिन के बाद इंडिपेंडेंट मिला है मैडम के सामने नहीं है तो
पहले देखिए पहले कल आए कल आ गए हम पहले 2 डिपार्टमेंट के हैं डीन फिर एडिशनल 2 दिन चालू थे तो पागल हो गया था तो लोग सर अपने साइन करके ये तो हम लोग तो ऊपर चेंज किया हाँ वही है आंटी बड़ी लेके चलो इसमें दिमाग खराब हो जाता है सब लोग अपना वगैरह तो मैं क्या करता हूँ पहले इतना मोटा फाइट इतना मोटा फाइट स्टेट्यूट्स ऑर्डिनेंसेस क्या क्या पूरा टैग करेगा तो विद परमिशन ऑफ चेयर एजेंडा नंबर 3 छोटी कुकिंग में रहूँगा मैं बड़ा मैं तो खाने वहाँ गए काजू कतली खाइए ये कल हाँ हो मुझे पता नहीं my note of dissent will be noted. So look how will the correction. जब हेड बनोगे
Hello, good afternoon, everyone. All the external participants, please collect your certificates from downstairs, from the reception. Hello, can you hear? Dilip. Dilip. Hello. चल नहीं रहा, ये निकालो यहाँ से, लैपटॉप वाला निकालोगे तब चलेगा। लैपटॉप वाला यहाँ से निकलेगा, स्ट्रीमिंग वाला, तब यहाँ पे इसकी आवाज़ आएगी। ऐसे ही तभी आई आवाज़ वैसे तो।
बोलिए। हेलो हेलो हेलो
हेलो।
नहीं है। बोलो।
हेलो।
बोलिए मैम।
हेलो।
आप बोलिए।
हेलो।
नहीं है। फिर से बोलिए, फिर से।
हेलो। तुम यहाँ खड़े होकर माइक टेस्टिंग करो, माइक टेस्टिंग नहीं करो।
आप बोलिए।
हेलो हेलो।
All the external participants are requested to get their certificates from downstairs registration desk at 3:40. Okay. Before that, we'll start our valedictory session soon. For the best oral and poster presentations, awards will be also given here. So be patient. Or meanwhile, you can go and get your certificates. External participants only from the registration desk. You can go and get it now, before we start the valedictory session here. And come back here, all of you. And come back here for the award ceremony.
हेलो हेलो।
Welcome back, all of you, and good afternoon to Honorable Pro-VC, sir, Registrar, sir, all the invited guests, faculty, and students. And a warm welcome to all of you to the valedictory section of the conference, International Conference on Advances in Cancer Biology Prevention and Therapy. Over the last 2 days, we had deep insights into several issues of cancer research like oncogenesis process, need of early diagnosis, personalized medicine, chemoprevention, inflammatory cytokines, and lifestyle changes required for the cancer prevention. Now I would like to call on the stage Professor Dinesh Raj Modi for the valedictory address.
A very good afternoon to all, respected Vice Chancellor, Registrar, and the invited delegates, Dr. Rath, Dr. Monisha, Dr. Sunil Babu, and other delegates, staff of the department, and also the students. Today, on the second day I came here, then I can realize what could have been the scene yesterday. I'm very happy the presentation, deliberation presented by the speakers was really what to say. It was really boosting the mind and all that, and they were all talking to the current topics. Okay. Nothing the old one, they discussed the matter and the interaction we had. And the other experience to me today, while interacting with the presentations, okay, and all the speakers over there who represented, no doubt they were the students for different universities and all that, institute and all that. But they all had the relevant latest topics, okay? And they tried their best to present the things in the best possible way. Not last but the least, I submit my apology mostly to the students and most of the delegates were there. I was responsible for them for missing the lunch, okay? Because when I got inside into the presentation and all that, so naturally there should be some questions over there. So then only there will be interaction and all that. And then I think that was one of the reasons why we were delayed by 15, 20 or half an hour. Because those candidates who are presenting, they should not have a feeling that it is a simple cakewalk, just making a formality and then moving out. Because it is not in any political stage. This is an academic stage. Okay, when we interact, the subject should be discussed properly and there should always be interaction. Question about and everything will be there. Okay. So what I experienced today over here, still I'm just telling you not to just pose something. Eh? You must be seeing for my face and mind, I'm still feeling fresh because mentally you are satisfied. Okay. The physical thing is secondary. First the mental thing is satisfied, then the physical things comes. Okay. So I never felt because like thing it is being delayed or something like that and all that. So most of I should not comment, most of the time it so happens, such as scientific deliberations and all the academic ones, the people just take it as a formality, come attend, meet few of their friends, and then move back. And another very happy occasion for me is that I think after 12 years, I was meeting Dr. Rath. Okay. Then we exchanged our experiences and also with Dr. Monisha Madam also. Okay. Though we were having the contacts on the physically, okay, The first thing was the 2 years because of this pandemic COVID. The movement was restricted and all that. And then, after the presentation of the students, myself, Dr. Monisha Madam and other colleagues were discussing, these 2 years, the batch which has passed out, we did not see them face to face. So, and for this was the batch where the students' interaction was not there, and then everyone expressed their experiences and all that. So overall, I congratulate to the organizers and Dr. Tabish and all his team, okay, that is, they have arranged everything. So it was in the spirit, it should be a scientific one. Okay, today I have gone, listened and attended the presentations. It was excellent. And then immediately after coming over, I see the best judges will not be from the organizers. So I interacted with few students. Then I asked him, क्यों भाई सब ठीक रहा? अरे नहीं sir, बहुत पता चला अच्छा interaction हुआ है. Because they are the ambassadors, they will give me the feedback, not the others. Okay, they are the right one to interact. Then sir, अच्छा रहा, interaction हुआ है and all that. And one more thing, I think this is after a long time, earlier to COVID was once my visit when this institute was under development and all the buildings. And after that, if I rightly recall, it was online in the Board of Studies meeting. Okay, I had that also. After that Board of Studies meeting, one day, one morning of the day, that was in the department, Tabish came to meet me, and as usual, it was. Then he gave me, that is, Dr. Sahab, online रहा, तो हमारे institute से आपको momentum भेजा है. Okay, I took that in momentum. Okay, and not immediately after 2 or 3 days, then I drew the attention and then I opened that. It was a canvas painting. Okay. Then I unfolded it, giving the historic buildings of this place of Lucknow and all that. Immediately, the only word from my wife and both of my son, Papa, इसको जल्दी से frame कराइए. So I told my younger son, Beta, I have no time. Then he took the responsibility, got it framed. And that is being displayed in my drawing room over there. And whosoever guest comes, they get surprised. अरे भैया इतना अच्छा कहाँ से मिला? नीचे दिखा है. बहुत सारे लोगों ने एडवाइस किया था नीचे से काट के फ्रेमिंग करिए. No, nothing. Originality will be there because I got it. Okay, and I don't depart with the gift. Okay, even some of the gift given during my schooling, then all the course, I still maintain it. Okay, gifts are not the value of money, it is the emotion attachment. They don't have any such element. So that's why I got it. It is there in my drawing room, and whosoever the visitor who comes and over there, अरे भाई साहब आप कहाँ से लेके आए, कौन सी दुकान पर? नीचे पढ़िए वो. Because it is at a distance, they don't read the what is written. Then they go and then they say, अच्छा अच्छा ये इंस्टीट्यूट से मिला है, क्या वहाँ पे एग्जीबिशन था? बोला भैया एग्जीबिशन नहीं था. Then I tell, because they are not from this profession, that the normal. Then अरे आपने बहुत संभाल के रखा है. हाँ बहुत अच्छा है. बोलते that is historic building of city of Lucknow. Na? If I rightly remember that it is Bada Imambara, Chota Imambara and Rumi Gate and all that. That is a canvas painting, not a printout. Okay, by an artist. So from that point only, I had, not to exaggerate, I had a feeling that this institute has some concern with academics, science and everything, and they are proceeding with it. You can just ask Dr. Tabish, I will be talking to him, क्यों भाई कैसा सब ठीक चल रहा है, कोई दिक्कत तो नहीं? नहीं सब बहुत बढ़िया है, ऐसा ऐसा चल रहा, हम ये कर रहे हैं. Then he called me that we are organizing a conference. Then first I told him, you just tell me the dates. And it was so lucky for me because 23rd and 24th I was busy of some internal work. I told him, then I saw, again asked me, please send me the itinerary. And then I told him, beta, हम 24th हम आ नहीं पाएंगे, 20 ऐसा है, okay. Then I was coming. So again, not taking much of time to you, but I really from my heart I tell you it was a very successful, not the word, a superlative to that could be. Okay, I'm not able to add, it is not in my vocabulary now, that we say good, better, best. Okay, so about that it was. Okay, the representation and the way the proceedings went on. Okay, and in future also, I wish you all will be continue this tradition, because, and I inquired from where are the students employed and what are they, because my curiosity is to know after this where the students are, what about the placement. Then they say they are mostly, see that is a success wherever you go, है ना. जैसे घर में आता, अच्छा भाई फैमिली ठीक है, बच्चे की तो क्या कर रहे हैं, बच्चों के बारे में पूछेंगे, पैसा नहीं पूछेंगे कोई कि आपके पास कितना पैसा है, कितनी प्रॉपर्टी है. They will be asking सब लोग कैसे हैं. So my nature is the moment any institute I go, the moment I don't interact with the administration or the faculty. I interact with the students, okay? Because they are the ambassadors. They will tell me what it is, okay? This is it. And I'm very happy, especially with the students also. I wish all the students and best of luck for them for the future ventures. And also I am again thankful to this organizing body and especially to Dr. Tabish and his group for inviting me for this occasion. Okay, I am highly delighted. You can just know from my feelings. I've been telling, अरे ताबिश हमारे तरफ से बहुत सारे लोगों का लंच छूट गया है ना. So कोई बात नहीं सर, ऐसे होता रहा है. तो he is used to me. हम कहीं भी जाएंगे एग्जामिनेशन जैसे सर दो घंटे का एग्जाम होगा, मगर वो 5 घंटे हो जाता है. ओके, तो ठीक है भाई, हमारी आदत पहले से है कि जिस काम के लिए आए वो करना है. बाइक का तो चलता रहता है ना, जस्टिफाई, क्योंकि हम ही लोग जा कर के फिर बाद में जस्टिफिकेशन के बहुत भाषण देते हैं. जब अपने पे करने आता तो कुछ नहीं करेंगे, वो नहीं करना जो हमारे गुरु लोगों ने कोई सीनियर से जो सिखाया है, हम लोग वही आगे लेके जा रहे हैं. सो विद दिस फ्यू वर्ड्स आई विल टेक लीव and thank again to the organizers and thank you very much.
Thank you, Professor Modi, for your kind words. Now for the award ceremony for oral and poster presentation for the conference, I would like to invite Honorable Pro Vice Chancellor, Sir, Professor Dinesh Raj Modi, Sir, and Dr. Sunil Kumar, Dr. Siddharth Vats, and Dr. Tabish Kidwai, convener of the conference. So in the oral presentation category, the first prize goes to Haram Sarfaraz from Integral University. And the second prize goes to Ms. Sonal Prasad from SRMU. Third prize goes to Saurabh Kumar from Lucknow University. And in the poster category, the first prize goes to Pransu Srivastava from SGPGI. Second prize goes to Yusuf Saleh from the Integral University. And the third prize goes to Prankur Avasthi from Amity University. Thank you, dignitaries and the participants, for the nice presentations. And now I would like to call on the stage Dr. Gurminder Kaur for the vote of thanks.
A very good evening to Honorable Vice Chancellor Sir, Pro Vice Chancellor Sir, Registrar Sirs, all the faculty members, session chairs, speakers, and participants who devoted their valuable time for this international conference on advances in cancer therapy, prevention, and in therapy. First of all, I would like to express my sincere thanks to Honorable Chancellor Sir, Honorable Pro Chancellor Ma'am, for providing us this platform for organizing this conference and for their constant motivation and support during the entire conference. Furthermore, I would like to express my thanks to Honorable Vice Chancellor Sir, Honorable Pro Vice Chancellor Sir, and Registrar Sir for their valuable advices and support during the entire conference. I extend my thanks to the eminent professors, scientists, and all the participants for being a part of this scientific event. Further, I would like to extend my generous thanks to all the advisory board members for their finest suggestions and great scientific inputs. I would like to express my sincere thanks to our chief guest, Professor Gaurav Agarwal, who accepted our invitation and shared his great experiences related to breast cancer, and also providing us the finest inputs in the field of cancer. I would also like to thank Professor Dinesh Raj Modi for his valedictory address and also for motivating our students. That was very important. I really liked the way you were asking the students. This shows that you have a very good interaction with the students, and I'm really thankful to you for that. Now, I'm highly thankful to our speakers and session chairpersons and co-chairpersons for being a part of this conference. I'm thankful to our sponsors, Experium, Times, BioVerse, Cytogene, and Vishal Hospital for supporting us in organizing this conference. I'm thankful to our technical team and from media department, from mechanical department, and also from diploma, especially Rohit sir, and system admin for making the live streaming and also the online sessions very thankful. Now, further, I would like to thank all the members of IBST for Faculty of Biosciences and Biotechnology who put all their efforts in carrying out their paths. Actually, the paths were given to all the committees, and they worked really hard for making this conference a grand success. Now, last but not the least, our students. A big round of applause for our students, and also without their support, without the support of our students, nothing was possible. So big thanks to the students of IBST and also for our other departments who helped us in making this conference a big success. I would also like to acknowledge our store department, registrar office, and deputy registrar admin for their cooperation. My special thanks to all the workers who put all their efforts in cleaning, in organizing the lunch, the teas, and everything, and making all the things. I would also like to thank our club students for participation in our inaugural session. I'm fortunate to thank all the dedicated and well-motivated team of SRMU organizing committee who were aware of their task and also their dedication to make this conference a grand success. So last but not the least, thank you very much. Thank you all for making this conference a grand success. Thank you.
Thank you, Gurminder ma'am, for the vote of thanks. Thank you all for joining us for this conference, and I hope you all enjoyed your time here and discussions. I hope we will meet again soon in some other conference. Thank you. And everyone, please join us for the tea. Before leaving, join us for the tea. And come, all the participants, please come here for the group photograph. Yes, sir.
बेटा ये याद रखो, ये प्लेस इज नोन बाय द पीपल। लखनऊ वगैरह यहाँ के रहने वाले नवाबों के तरफ से आ रहा है, ऐसे ही इंस्टीट्यूट रहेगा स्टूडेंट्स के तरफ से।